Uh, Ladies and gentlemen, good morning in Thailand, and maybe I have to say good afternoon in Japan. So welcome to the 7th Priority University Symposium. My name is Nopollet Vattanaskun. I come from Department of Microbiology, Faculty of Science, Kasetsart University. So today I will be your MC. So now I think it is time for the opening ceremony. May I invite Associate Professor Dr. Sutkhet Nakasathien, Vice President for Research and Creation, Kasetsart University, to deliver an opening address. So please, Associate Professor Dr. Sutkhet Nakasathien.
Welcome to the 7th Priority University Symposium. The role of research technology in stabilizing against global disruption. On behalf of Kasetsart University, I would like to thank the lecturers, faculty members, students, and all participants. Thank you very much for your kind cooperation and for attending and participating in this symposium. This symposium has come a long way since KU launched the first symposium in 2014. Kasetsart University and Yamaguchi University have a long history of friendship. We would like to express our sincere appreciation to Yamaguchi University for its great support and cooperation over the past 2 decades. In November 2016, Yamaguchi University, Japan, established International Collaboration Office of Yamaguchi University, YUICO, and the Faculty of Science, Kasetsart University, Bangkok. Additionally, in 2018, the Faculty of Agriculture and the Faculty of Science of Kasetsart University entered into a cooperation agreement with the Faculty of Agriculture, Yamaguchi University, to create a joint course leading to joint master's degree program in agricultural science, in agricultural and life science. This program was launched and accepted the first class of students in academic year 2021. We will now have our first batch of graduates in March of 2022. Recently, in December 2021, BCG Chulalongkorn University, Yamaguchi University, Japan, and Silpakorn University organized an international workshop in order to exchange information and opinions regarding biotechnology and engineering. We gladly appreciated the opportunity to exchange perspectives with the participating universities. The program covers a wide range of topics and challenges that are highly relevant to all of us. Those discussions will undoubtedly prove beneficial to us in our ongoing efforts to develop our research, pursue innovation, and strengthen our educational initiatives. Finally, I would like to thank you to all of you once again for attending. I invite and encourage everyone to participate actively by sharing our expertise, knowledge, and ideas. I wish all of you an enjoyable and rewarding symposium. Thank you so much.
Okay, thank you very much for your wonderful message. So I will move to the next session. To all participants, you may download the abstract book posted on the chat board. So next, may I invite Mr. Masahiro Ueda, Chief of International Affairs Division, Yamaguchi University, to give a talk in the title of Introduction of Priority University. So if you are ready, please start.
Okay, thank you for having me today. Let me share my slide. Okay, can see me?
Yes, you can start. Yes.
Okay. Hello, everyone. My name is Masahiro Ueda. I'm Chief of International Affairs Division, Yamaguchi University. And now I'll be talking about our priority universities program, which is one of the core program for our international strategy. Yamaguchi University celebrated the 200th anniversary of its founding in 2015. And at that time, we formulated the University Vision 2015 for the next 10 years. In the vision, we set up the slogan, create a diversity campus through the 200th anniversary. We have aimed to create a diversity campus where every member accepts the diversity such as ethnicity, culture, language, religion. Then new values are generated. Under the slogan, we have been advancing internationalization, both in education and research. We established the Faculty of Global and Science Studies in 2015. In the faculty, 100 Japanese students go to study abroad, and 100 foreign students come to Yamaguchi University as exchange students via its curriculum every year. On the other hand, in order to promote international joint research, we have designated several overseas partner universities as priority universities for cooperation. And we provide support for international cooperation activities with these universities. We see great potential for the university's research level to grow through connection with these universities. And we have set goals of providing educational support, contributing internationally through academic exchange, and increasing the overall level of research by strengthening research-focused international cooperation activities. 3 years after the launch, we made interim assessment of this project, and after the 5-year term, we will make post-project assessment. At present, we have had 12 priority universities for cooperation. Especially, we have designated Kasetsart University and Choowongkomon University since 2013 and built strong relationships. This slide shows the outcome of the priority universities program. Over 100 international co-authored papers have been published, and research presentations have been conducted at many international conferences. In addition, researchers have been meeting actively. Unfortunately, we can hardly carry out international activities for the last 2 years due to COVID-19, but we can see the program is accelerating international research cooperation. Most of the international co-authored papers come from this project, the development of tropical environmental microbe resources. I think the reason is that many researchers from various fields are forming teams and participating in the project. I'm sure organization-to-organization relationship is the key of accelerating international research cooperation. The current program is going to finish this March, and we have started to make a discussion for the phase 3 of priority programs. We are considering keeping most of the ongoing project in the next phase and developing them by promoting organization-to-organization relationships, referring to the project with Kasetsart and Choowongkomon. In addition, we also encourage a co-creation education program into every faculty in Yamaguchi University. In terms of the situation for national universities in Japan, the fourth midterm goal period is going to start this April. We will commit to creating a diversity campus from the aspect of internationalization by carrying out the priority universities for cooperation program and co-creation education program. We are grateful to work together with you from now on as well. That's all. Thank you for listening.
Okay, thank you very much, Mr. Masahiro Ueda, for your important introduction. So next, we will move to the next speaker. Next we are going to have a group photo in a virtual version together. So may I ask all of you to open your camera for a while, please? Okay, all participants, could you please open your camera for a while, please? So our staff can manage to capture the photo together. And maybe I will count down from 3 to 1, and maybe you can smile. Okay. Okay, 3, 2, 1, smile. Okay, just remain for a while because we have many participants, so we may capture many screen. Okay, our staff, can we go to the next session? All right, I think maybe the photo has been taken. Thank you very much, everyone, for your kind cooperation. So next, may I invite, maybe I will go to the next speaker. May I invite Associate Professor Dr. Sutkhet Nakasathien, Vice President for Research and Creation, Kasetsart University, to give a talk in the title of Reinventing KU, Reinventing Integrated Research Collaboration. So, Vice President, please start if you are ready.
Thank you so much. Could you please share the slide? Share slide ได้เลยครับ Okay. Call full screen now. Good morning again, ladies and gentlemen. I'm very happy to be with you this morning or this afternoon in Japan, talking about Reinventing Kasetsart University. This quite new program of all Thai universities, and Kasetsart also participate in this program under the Ministry of Higher Education. Certainly, under the Reinventing KU, we also have to reinvent our international research and collaboration that I would like to share with you this morning. By standing of Kasetsart University at our 80th anniversary, I have to congratulate to Yamaguchi that you are stepping towards the celebration of your 200 years this soon. And I just want to share the information that by QS World Ranking by subject of agriculture and forestry, the standing of Kasetsart University is like up and down in the range of 29 to about 60 in the past 6-7 years. With this kind of changes, we have to rely on index that has been used to measure the performance of the university in the big picture. Last year, Kasetsart University stands in the 63rd in agriculture and forestry that I would like to share with you. With this kind of situation, how can we maintain or improve our standing in the world rank level? Not only in academic, all the university is in Thailand, and I believe that all the university in the world also have to be challenged in so many platforms of ranking, as well like in the SDG that Kasetsart University also emphasize on this, because with the 17 goals of the SDG, we also have to focus in what we are good at. And by Kasetsart University, we chose the zero hunger, life below water, climate action, and partnerships for the goals that can be changed, can be modified year to year. In the overall picture, Kasetsart University have all kinds of tasks in academic, in research, in academic services, or in culture ways that we also have to follow the protocol of the university. Under the Reinventing University and Research Institute of Thailand, we have so many things to do. I would like to share with you, like first of all, about the manpower and knowledge development. We have to focus on improvement, not only on the faculty member, researchers, scientists, graduate students, postdoc, covering undergraduate students, onwards to the lifelong learning and future skill sets that we also have to do that. Secondly, for the research development and innovation for Grand Challenge. When we are talking about Grand Challenge in Thailand, we are talking about the platform. And thank you, Yamaguchi, so much. That has been a very wonderful friend for Kasetsart University that we have been working together. And not only Yamaguchi University, we have to work with so many universities worldwide. And for research and development and innovation competition, all kind of new platform, new technology, advanced technology, all kind of support, not only on the infrastructure, the physical, and also on the human resource development. And not only on the advanced part or side, we also have to work with the localities, the local economy, the innovative community, also the focus of the university. Recently, the university system in Thailand has been classified into 5 groups, and Kasetsart is in group number 1, that are classified as a global and frontier research university. For other groups, it can be said that, for example, for technology and innovation in the second group, for area-based and community, for this group, for example, for the area-based, going to be other universities that focus onto their provinces or their region, and the moral and intellectual cultivation, and as well for the specialized professional university. So with this classification, different universities have different focus. And Kasetsart University is in group number 1. We have to do like advanced science by also maintaining the service to the local. By having this program starting in 2021, the focus is in agriculture, food, fisheries, and forestry. And not only that, we also have the second layer of environment, science, engineering, medtech and wetech, and as well related to economics, education, and social science. With this, we have 3 different, we have 3 purposes. The first one is for research capacity strengthening, with the involvement of more than 1,000 potential researchers in the next 5 years, and also trying to come back in the 50 ranked of the university by the subject of agriculture and forestry. And we need to create or support more the world's top 2 scientists. Now we have some, but we need to have more. And by running the international collaboration, we expect to have more of the 20%, top 20%, top 10%, top 5%, and top 2% in different areas. And we have different programs. The first one is for the outbound joint research with world-renowned strategic partners. That means we have to send our faculty members, researcher, postdoc, postmaster, PhD, and undergraduate. And we can do about 120 of them in the past year. Even under the COVID-19 situation, it is quite difficult for Thai university, or I think the university around the world, that we are facing this situation. And for teaching and research and development, we also have the inbound program by inviting the famous professors to work at KU. And also we organize international conference. I think more than 20 conference has been done with the goals that we want to improve the H index in the big picture and also the university ranking in Asia. Under this program in our first year, we sent 83 scholars with the outbound program to America, to Europe, to Japan, and as well to Africa. Some of them still stuck in Thailand under the COVID situation, but we are trying to okay, back to this slide. By the history of our KU and Japan research collaboration, by paper, we have 115 collaborative programs with different institutes. And we have so many very active. For example, I was a former dean of the Faculty of Agriculture. Each year, we have more than 20 active programs with the university in Japan by the Faculty of Agriculture alone. If we count all the faculties or research institutes under KU, we have more or many active collaboration. So I think we have to keep going this way with the Japanese university and between the government of the two countries. Looking into the records of the publication, we are good in different ways. I don't want to compare or benchmarking, but I would like to share with you or leading the idea, but what we can do together. Yamaguchi University is very good at medicine, while Kasetsart University is very good at agriculture and biological science. With the number of publications, Kasetsart University has about 3,000 researchers comparing to Yamaguchi University. I think I'm not sure that how many, but we still have to support or push more on our researcher to do more work. And by having the outstanding fields, I think this can lead to our ideas in the future that what we can do between our two universities. In the past, I have to thank to our professors that has been working so hard in the past 2 decades. I saw Dr. Kanchana also online this morning. Thank you so much, Dr. Kanchana, for joining us this morning. She is the one of the key professor who took initiative and also keep running all the programs. With the time limitation, I will not go into the details of each picture, but I would like to share with you our record of collaboration and all kinds of work that has been done together through the CCP activities and number of projects, the 5 projects with the output. Mainly it's going to be focused in microbiology, but we expect to do more work, especially under the joint degree program we are trying to expand into like biological science or other fields. Some of the accomplishment between KU and YU collaboration with the publication with different strains of the microbes that has been worked through the collaboration of the university. I think it is our highlight that it can be marked. Also, we have a current student in master degree in life science, Mr. Nakaki and Mr. Tony Oka. That thank you so much for working so hard, and I think that they're gonna accomplish more and more in this near future. And for the highlight on the synthetic biology, that the cutting edge technology, we have two good publications that working together. And in 2018, when I was a dean of agriculture, I also get a chance by led by our president, Dr. Chongrap, and our team that we went to Yamaguchi. It was fun. I remember all kind of research idea exchange and symposium. And I would like to focus in the second part on the Reinventing international research collaboration. Under Kasetsart University, it can be said that with the emphasis on the biological science, the physical science, the future energy, and also technology that I'd like to share with you today. We have one of the highlights of the program that working closely with the Japanese university, the development of duckweed holobiont resource with all kinds of, with many aspects of collaboration. For example, the functional food, the bioplastic, or the future food, animal feeds also been done. And our key partner is in Hokkaido. And not only in Hokkaido, there are so many universities in Japan and also in Thailand. This is very big platform that had been supported by JICA. That I already mentioned that about the animal feeds. It's going to be like the hidden treasure or the hidden natural resources that we have to focus and work together to identify. Recently, we talked to NASA of the United States. It might be another candidate project that's going to be working with NASA for the future food for the people who are working like in the space. And also duckweed can be used for wastewater treatment and for the greenhouse gas mitigation. And recently, by the experience of Thailand, we have a big problem of the animal diseases caused by the virus called African swine fever and as well PRRS, covering that happened in the pig farms. I think nearly half of the population of the pig that has been affected by the disease. That is a big problem, not only in Thailand, but in Asia and also in other countries. And with the animal genetics and genomic research, they are focusing on improving the genetic resources in a wide range of the species, not only on the conventional method, but also on the genomic technology. And also the animal feed production by the classical way, about the formulation, about the developing or finding the alternative of the animal feeds. They also focus on microbiomes of different species that whether or not they can identify what can be useful for the animal species. For the functional food ingredient, also have another team in Denmark and also in Japan. And by the development of the system biology and microbiome, we are also working in the big picture, developed the platform at the national level with about 13 university and research institutes and private sector working together under this platform. And also the future material, the cellulose and future materials It is a close work together with the university in China, Dr. Lee, and also in Austria. And for the medical entomology, we have of the world-renowned scientist, Dr. Thilapap, who are working closely with the French team, the USA researchers, to eradicate the dengue and malaria in tropical areas. So I think this might be the field of interest for the medical researchers to work with KU more in this field. And synthetic consortium that I already mentioned. And for the another part for the physical side, with the collaboration with NASA, we just have our first MOU with the NASA last week by developing the liquid crystal to be used in the space, like in the helmet or in our cell phone. It's going to be the game changer in the future. And also, we will be the part of the flight operation called Oasis 2 in 2024 that we're going to send our experiment with the flight. And for the big data and analytics as well, I think this is a hot key area because we have gathered information a lot, not only in agriculture, but in other science, and also with the advanced technology for the drone picture, the satellite picture, the genomic data to support for the biodiversity and as well for the either plants or animal improvement program. So we have received the big funding support by the PMUC under the Ministry of under the government of Thailand, that we will develop the digital data platform starting this year. And lastly, I would like to mention a little bit about the sustainability. Whatever that we have been doing, we have to realize about the sustainability either by the human or in the environment. So SDG goes everywhere, and I believe the similar situation also happens in Yamaguchi University. And with the 17 goals, and we are focusing on 4 that I already shared with you, and to work together in this since the past up to now and towards the future. Very much Kasetsart University would like to keep our friendship and also sending it forward to our future generation to create value-added work and more creative pieces of work together. Thank you so much. That's all my talk. Thank you.
Okay, thank you very much, Associate Professor Dr. Sutkhet Nakasathien, for your remarkable presentation. So if some participant, if you have any question, maybe we still have time to ask some question.
Yes, I would like to invite the audience to share with me, or if you have any questions or suggestion, it's going to be useful for us. And by the fields that I emphasize today is only for the fields that related to the Reinventing University. Actually, there are so many more interesting work. I saw Dr. Patchareeya in the audience that she's gonna talk about her work this afternoon that emphasizing on the orchid, I guess, right?
Yes, right.
I think that in this near future, we will share with you more on the other field that we also have been focusing on.
So is there any question from the audience? You can grab the mic and ask the questions or comments.
Under the COVID-19, it has been 2 years already that we have not seen each other in person. All the friends that have been acquainted at YU, I think hopefully in this near future we will meet again. Dr. Kanchana, would you like to go to Japan this soon? I saw you in the audience. Yes, you are there. Okay. Yeah, I think there is no question.
Okay, if no question, thank you very much. Thanks again, Associate Professor Dr. Sutkhet.
Thank you so much for your invitation.
Okay, next session, the scientific programs. We have today 8 oral presentations by the representative from 3 universities, 4 speakers from Kasetsart University, and 3 speakers from Yamaguchi University, and 1 speaker from Chulalongkorn University. So I would like to give some information about the times. So each speaker has 20 minutes for presentation and 5 minutes for question and answers. So speaker will be notified 1 ring after 18 minutes pass, and 2 ring when the time is up. So please be notified. Okay, I will start from the first speaker. May I invite Professor Dr. Hiroyuki Azakami from Yamaguchi University to present in the title of several approaches to biofilm control. So Professor, if you are ready, please share your slide and start presentation.
Thank you very much for introducing for me. Okay, I share my slide. Is it okay?
Okay, we can see your slide.
Hello everyone. I'm Hiroyuki Azakami from Yamaguchi University. Last November, I organized a Young Scientist Seminar. Thank you for the participation and cooperation at that time. This is my first time to participate in this symposium. I'd like to thank everyone involved for giving me such an opportunity. Today, I'd like to talk about our study approaches to biofilm control. Some of bacteria are planktonic. They floated around in water. Since the time of Pasteur, many microbiologists have studied using a suspended bacteria culture. But most of bacteria in nature are attached to surface and they lived in biofilm. Bacteria living in a biofilm can have significantly different properties from free-floating bacteria. One benefit of this environment is increased resistance to detergent, enzyme, immunity, and antibiotics. In the community, bacteria are communicating each other. Instead of language, bacteria use communication signal molecules released into the environment. These molecules are called as autoinducer. By releasing this signal molecule, bacteria are also able to measure the concentration of the molecule within their population. This figure shows example of communication signal autoinducer. In gram-negative bacteria, there are two major autoinducer, AI1 and AI2. AI1 is a acylated homoserine lactone, AHL. It is used in intraspecies communication. In contrast, AI2, this is this mediates intraspecies and interspecies communication. On the other hand, gram-positive bacteria, they use the cyclized peptides for their communication. Bacteria behave quietly when their friends are not around them. However, when number of friends increase, whole bacteria group begin to behave badly. Activated bacteria can express virulence factor or toxin and home biofilm. If they start doing bad manner with few friends, they will be found and killed by host immune system. In our laboratory, we have studied several approaches to biofilm control. One is biofilm inhibition by food-derived components, such as tea catechin and mushroom extract. The second is inhibition by quorum quenching. First, I'd like to talk about biofilm inhibition by food-derived components. In our study, we used this microbe, Eikenera corrodens. This is one of the periodont pathogenic bacteria. E. corrodens is a gram-negative facultative anaerobic bacteria. These are structures of catechin, 8 catechins used in my study. We tested the effect of 8 catechin on biofilm formation E. corrodens. As shown in this figure, some of catechin inhibit biofilm formation. Next, we tested the effect of different concentration of catechin on biofilm formation. Some catechin inhibited biofilm formation below MIC, minimum inhibitory concentration. For example, EGCG epigallocatechin gallet that is inhibited biofilm formation at 0.1 millimolar concentration, whereas EGCG MIC is 0.5 millimolar. At this concentration, catechins inhibited biofilm without antimicrobial activity. Next, we tested effect of catechins on biofilm formation of lactase deficient mutant. This strain cannot produce autoinducer AI2 and cannot communicate each other. Against lactase mutants, low concentration of EGCG did not inhibit biofilm formation. This is wild type. This result suggested quorum sensing bacterial communication is involved in biofilm inhibition. In summary, we showed that biofilm formation was inhibited by some catechin at low concentration without antimicrobial activities. Structure of catechin was important for biofilm inhibition. Moreover, we suggested that bacterial communication related with biofilm inhibition by catechin. Next, I'll talk about biofilm inhibitor from mushroom extract. In this study, we selected 6 edible mushrooms. These fruiting bodies were extracted with water or methanol, and their biofilm inhibitory effect was examined. Among these extracts, biofilm inhibitory effects were observed in the water extract of these 3 mushrooms and methanol extract of these 3 mushrooms. First, we studied methanol extract Shiitake mushroom. Shiitake is one kind of edible mushroom. Pharmacological effect on tumor and diabetes have been reported. In its water extract, antibiofilm activity has also been reported. First, we compared fresh shiitake with dried one. This is dried shiitake. Biofilm inhibitory effect was found in dried one, but fresh one showed more stronger inhibitory effect. Next, we examined the effect of the number of days stored at room temperature. Inhibitory effect was lost 5 days after storage. Next, fruiting body of shiitake were divided into the pileus part and the stem parts, and their inhibitory effect was compared. There are also inhibitory effect on stem parts, but pileus part showed the more stronger effects. When inhibitory effect was compared by changing the amount of extract added, methanol extract of shiitake mushroom inhibited biofilm in a dose-dependent manner. Furthermore, when amount of shiitake extract showing the sufficient biofilm inhibitory effects was added to the bacterial medium and culture. As shown here, no inhibition was observed in the growth. This suggested biofilm inhibition by shiitake extract is due to the mechanism other than antimicrobial activity. Bacterial communication has been reported to be involved in biofilm formation in several cases, including T category. Therefore, I created, constructed a lactase deficient strain that cannot communicate. This is a biosynthetic pathway of AI2 autoinducer in the E. coli. Thus, this lactase enzyme is necessary, essential for AI2 biosynthesis. So, lactase deficient, we knock out this enzyme, and resultant strain cannot this yellow color cannot produce AI2, and so this strain cannot communicate. We compare shiitake. We compare the effect of shiitake extract against the two strains, wild type and lactase mutant. Wild type strains are more sensitive to inhibition of shiitake extract than lactase mutant. This result suggested bacterial communication may be involved in biofilm inhibition by shiitake extract. So, next, shiitake extract was added to culture medium of E. coli, and the AI2 amount was compared. The results showed addition of shiitake extract reduced the amount of AI2 production by E. coli. This suggested shiitake extract may inhibit biofilm by inhibiting bacterial communication. In summary, it was suggested that methanol extract of the pills portion of fresh shiitake mushroom inhibited biofilm formation as a result of inhibiting bacterial communication. Next, we studied the water extract of the Yamaguchi mushroom. The Yamaguchi is also one kind of edible mushroom. Pharmacological activity of anti-dementia, anti-inflammatory, and antioxidant properties has been reported. Water extract of Yamaguchi inhibits the biofilm, but methanol extract has no activity. Next, Yamaguchi water extract that was fractionated with ammonium sulfate precipitation and check the biofilm inhibitory effect. As a result, inhibitory effect was observed from the 50% and 70% of ammonium sulfate precipitation fraction. So when inhibitory effect was compared by changing the amount of added extract, water extract of Yamaguchi mushroom inhibited biofilm in a dose-dependent manner. And like the shiitake extract, Yamaguchi extract did not show growth inhibition. Therefore, it was suggested Yamaguchi extract also inhibit the biofilm by the mechanism other than antimicrobial action. After heat treatment, Yamaguchi extract biofilm inhibition was examined. As shown here, inhibitory effect even was shown even the after heat treatment 50 degrees centigrade and 60 centigrade, and even after boiling remained partially. This suggested biofilm inhibitor in the Yamaguchi that is a heat-stable protein. So next, extract was fractionated by ultrafiltration and check the rough molecular weights. As a result, it was suggested that biofilm inhibitor in the Yamaguchi that is a protein of about 30 kilodalton or more. Biofilm inhibitor were partially purified from the extract by the two types of the ion exchange chromatography. As a result, inhibition of biofilm inhibition was observed depending on the protein concentration of this protein, 55 kilodalton protein. So in this area, two protein band was observed. So we analyzed the N-terminal amino acid sequence, these two bands. As a result, it was clarified that upper bound is glycosidase, and the lower bound is lactase. Next, we investigate whether extract has the ability to destroy the biofilm once formed. Biofilm destruction was observed in this figure, as shown in this figure. Biofilm destruction was observed within the 15 minutes after addition of the extract. Therefore, it was found that Yamaguchi extract also has the ability to destroy the mature biofilm. In summary, biofilm inhibitor contained in the water extract of Yamaguchi that is a protein of about 55 kilodalton with a heat-stable structure. That maybe that is either glycosidase or lactase. In addition to inhibition of biofilm formation, Yamaguchi extract has ability to destroy biofilm once formed. Next, I'd like to talk about biofilm inhibition by column quenching. What is column quenching? This is AHL, one of the autoinducer. In the case of AHL, two degradation enzymes are known. One is lactonase, AHL lactonase, and one is the other is AHL acylase. Lactonase hydrolyzes the this portion lacton ring, and acylase break the amide ring here. But both enzymes inactivate AHL by degradation. By the inactivation of autoinducer, communication was blocked. That is column quenching. E. colodens did not produce AHL, but as shown here, E. colodens produce AI2 autoinducer in mid-log phase. However, its growth reached the stationary phase. AI2 production decreased dramatically. So we expected that this effect is due to AI2 degradation enzyme. So we tried to isolate this enzyme from the culture supernatant of E. coloden. This is the result of AI2 inactivation assay. As shown here, 30% production of the ammonium sulfate precipitation. That this fraction inactivated AI2. So we expected AI2 inactivation factor is included in this fraction. So we try to purify further. Using the ion exchange chromatography, gel filtration, we purify the AI2 inactivation factor as a single band. This 40 kilodalton protein, we analyze this protein's N-terminal amino acid sequence and compare with the database. As a result, this protein is outer membrane poly. So next, we constructed Pauline gene deletion mutant and compare its AI2 production with that of wild type strain. As shown here, wild type strain pink color, wild type strain AR2 inactivation was observed in their stationary phase. But in the pollen mutant, this purple color, AR2 inactivation was not observed, even the stationary phase. This result suggests pollen is involved in AR2 inactivation. But in general, pollen is involved in membrane transport. So it is unclear how pollen inactivate AI2. Next, we added pollen, purified pollen, into the culture media, equal-dense culture media, and examine its effect on the biofilm formation. This is equal-dense biofilm. After addition of pollen, biofilm is inhibited. So this result suggests inactivation of AI2 inactivation by pollen, that is control of biofilm. Next, purified pollen was added to the culture medium of another bacteria, Streptococcus mutans. Streptococcus mutans is cardiogenic, caries bacteria. S mutants was reported that this microbe use the AI2 as one of communication signal. So as shown here, biofilm inhibition was observed. Pollen was added 18 hours after the start of culture. Furthermore, when pollen addition, amount of the pollen addition that increased, inhibitory effect was observed even after 12 hours and 18 hours. So as a result, it was shown that AI2 inactivation factor produced by E. clones that can act across the bacterial species and inhibit the biofilm formation of Streptococcus mutans. In summary, culture supernatant of E. clones at the stationary phase decreased AI2 activity. This suggested E. clones produce AI2 inactivation factor. 40 kilodalton protein was purified as a AI2 inactivation factor. N-terminal amino acid sequence suggested that this protein is outer membrane pollen. Purified pollen inactivated AI2 in a dose-dependent manner, and AI2 inactivation was not observed in pollen mutants. these suggested Pauline might be involved in AR2 inactivation in E. coloden. So now we expected that biofilm inhibitor will be developed by inactivating AR2 using the pauline. As you know, Yamaguchi University has a research center for thermotolerant microorganisms. From next April, I will be appointed as the director of this center. I would appreciate your support, including collaboration between three universities. That's all. Thank you for your attention.
Okay. Thank you very much, Professor Dr. Hiroyuki Azakami, for your interesting presentation. So now, I think now we still have time for about 5 minutes for question and answers. So audience, if you have some question, please maybe at the bottom of your screen, you can select the reaction button and also select the raise hand and we can see your name. Or if nobody, maybe you can just place the mic and start your question. Or another way also, you can type your question on the chat board and I will read your question for you. Any questions? Azakami Sensei, I have some question about the inhibition. I mean, the time for inhibition. I mean, the compound, I mean, can inhibit the biofilm formation for how long? I mean, impermanent or maybe temporarily?
Yeah, many inhibitors have been reported already. For example, polyphenol from plant or in mushroom, some enzyme.
I mean, the effect. I mean, how long the effect of the inhibition? I mean, is it impermanent or maybe temporarily? Can you explain?
Yeah, temporary effect. Not sorry, permanent effect. Mechanism is depend on each compound.
I see. So you mean also some compound maybe give the permanent effect, some maybe short time effect or something like that? Okay. Any more question from the audience? Okay, if no more question, maybe now the time is also up. Thank you very much, Professor Dr. Hiroyuki Azakami.
Thank you.
So we are going to have a short lunch break for about 30 or 40 minutes. So please come back to this room by 12:50 in Thailand times or 2:50 Japan times. So hope you enjoy your lunch time or maybe afternoon coffee break. See you next time. Okay, welcome back all participants for the after the lunch break. So hopefully you have enough lunch or coffee break. So we are going to the session 2. I will invite the speaker. The next speaker, may I invite Professor Dr. Kiattawee Choowongkomon from Kasetsart University to present in the title of Computer-aided drug discovery from small compound to protein inhibitors against tyrosine kinase of EGFR for cancer therapy. So Professor, if you are ready, please share your slide and start your presentation.
Okay, so good afternoon, everyone. So I hope you see my slide.
Yes, we can see.
Okay, all right. So my name is Kiattawee Choowongkomon. I'm from Department of Biochemistry at Kasetsart University. So today I would like to share with my research experience on last 15 years on trying to use the computer-aided drug discovery to identify the new inhibitors against the tyrosine kinase. So my target is I'm interested on the cancer, and from this slide we can show that the cancer will be the most problem in the world, and worldwide case of the in the cancer is seem to be increasing every year. So we expect that in 2030 there are going to be around 13 million people who died from the cancer, and the most important cancer in the world is the lung, breast, colon, prostate, and stomach cancer. And the most important thing that I'm focused on is the lung cancer. In the lung cancer, we found that 85% of the lung cancer will have the enzyme called epidermal growth factor receptor overexpressed on this type of the cancer. And this EGFR usually that's important for the cell signaling pathway. It's important for the cell proliferation, inhibit apoptosis, and important for the migration and the invasion cell. So the activation of EGFR is after EGF or growth factor receptor binding to the extracellular domain. It can cross the dimerization and can cause the signaling downstream in the cell. So therapeutic EGFR therapy by focusing on EGFR has been targeted for the several drug company. The first line of the drug is the monoclonal antibodies that bind to the extracellular domain, that including the cetuximab and panitumumab that has been using in the clinical right now. And another type of the drug is the small molecule inhibitors that target on the tyrosine kinase of EGFR, which are the gefitinib, erlotinib, afatinib, and osimertinib. That's my aim target, that I really want to find the inhibitors, the small molecule that inhibit on the tyrosine kinase of EGFR. So my research idea that has been trying to work all a long time is I just try to use the computer to screening the compounds, and then after that I test the compound in the laboratory. Another way around that we just try to do another way is we use some technique that's screening the compounds in the laboratory first, and then use the computer to answer the question why that compound is inhibited with EGFR. So that's both idea that we're trying to do in the lab for a long time. And my aim is I'm starting from the small molecule compounds, and then after that we move to the herbal compounds, and after herbal compound we move to test in the lab, like using the library of the cyclic peptide library and nanobodies, try to inhibit the same target of EGFR as a model in this case. So first of all is when I started the project 15 years ago, I looked into the NCI database, which is the database that's freely available and we can access that one. And they also prepare the diversity set of the compounds that we can starting with. In this database, they have the libraries of the compound that they have. They know that this compound can inhibit several types of the cancer cell. However, they don't know the mechanism of this compound. So my idea is if we can pick up one target of the enzyme in the cancer cell, and we know that this compound inhibit this enzyme, so we can know the mechanism of this compound inhibit the cancer cell via this enzyme. So this enzyme I pick up is the EGFR in this case. So that way is we start to do the docking. We preparing and make the protocol for the docking protocol. So in the docking protocol, we do both the orthodox and the go docking protocol, and from that to library, and we use the NCI dataset as the dataset. There's around 2,000 compounds at the beginning, and use the docking protocol or the orthodox and go docking score to identify the compound. With both methods, we found the consensus compound turned out to be 8 compounds after that. Of 8 compounds is the set of the diversity set at the beginning. So after that, we search to a similar compound in the database. We turn out to be 230 compounds available, possible to further test that similar structure from our 8 hit. So those 230 compounds we redocking again with another similar compound. It turned out to be 31 possible compounds after that. And at that time, we able to express and verify the kinase in the lab, and we set up the kinase protocol to identify the inhibitors by using the ADP flow assay. And when we developed the assay, that we can show that we can identify the IC50 against the gefitinib, erotinib, and lapatinib as a control compound, right? And we screening all the 31 compounds that we screen in the laboratory, in the computer, and we able to show that several compounds seem to be interesting result, and 2 of them seem to be show highly inhibition against the EGFR higher than another compound, which is the 116555 and 125910. Those 2 compounds, so we pick up several compounds and we just show few of them in this slide. The first one is 116555 and the second one is 125910. Those 2 compounds that seem to be screening from the computer seem to inhibit EGFR enzyme very well, even though it's around 6 times to 7 times higher than gefitinib. But then in the cell line, when we test in the cell line A549, which is the lung cancer cell line, right? So we found that in the gefitinib, we found IC50 around 64 micromolar, while the 116555 seem to be 10 times better than the gefitinib, which is 10 times least, only 6 micromolar for IC50, and 125910 is around roughly similar to gefitinib in this way. However, another compound, the last 4 compounds that seem to inhibit EGFR also, but it doesn't inhibit the cell line at all. That may come to that this compound cannot get into the cell. So from this, we can identify that 116555 seems to be potential new drug against the EGFR, which similar, which the structure is on the right hand side here is similar to the doxorubicin, right? So we also using the Western blot to confirm that our 116555 can inhibit phosphorylation of EGFR inside the cell, right? From this, from the figure 4, you can see that it can decrease the phosphorylation of the tyrosine 1173. And later on, we do apoptosis. We can show that this compound can cause the apoptosis inside the cell and can really inhibit the cell, cancer cell by apoptosis. And we show the docking, that how they interact with the EGFR inside the pocket site. That's the first part from the screening the small compound. Later on, we use the same protocol to identify the herbal compounds. In the herbal compounds, Professor Jak Sang Ma at Department of Chemistry at Kasetsart University, he developed the ChemiBase, the database for the Thai herbs, and we collect all the 30,000 compounds from the Thai herbs and get to the protocol, the same protocol that we do for the small molecule on the go and orthodox, and we turn out to get 8 compounds after that. So okay, we can skip this one. So those 8 compounds showing in this slide, right? So this is the source of the plant, like from the ebony tree, mangosteen, custard apple, legumes again, and white turmerics and walnut and neems. So this all structure look like. After that, we look at the structure of the compound. We found that at least 2 of them seem to be the sandtone structure. It's a sandtone structure. So from this sandtone structure, we gather the structure, the source of this xanthone derivatives. One of them we get from the Glacinia saphifolia that have the structure of the xanthone structure in there, and we test that compound. We found that one of them, compound 1, which have this structure shown in this table 4, it have the IC50 around 90 nanomolar, which is similar to gefitinib and can inhibit the EGFR very well based on this one. So we confirm that xanthone's compound or any xanthone derivative, it's possible to be the EGFR inhibitors also, and we published in the molecule in 2014. Later on, I give this talk to another university, and at that university, Dr. Park Poom, he got the compound, another xanthone compound called Choowongkomon that extract from the Glacinia cova leaf, and this Glacinia cova leaf had the structure of the Choowongkomon like this. So I decided to do some docking study and found that it seemed to be potential inhibitor against EGFR also. So we did the same thing that we check it on the cell line. This time we use the HeLa cell line. We found that the Choowongkomon can inhibit the HeLa cell line, which have the IC50 similar to alpha-tinib, which is the third generation of the EGFR inhibitor, which around 3 micromolar, which is the alpha-tinib is around 2 micromolar. It's really similar and better than gefitinib. Later on, we show that this Choowongkomon really inhibit the EGFR by got the IC50 around 3 nanomolar, which is better than the gefitinib and also similar to alpha-tinib that we found exactly in the cell line, right? And the docking score, the docking conformation show that it can inhibit block into the active site of EGFR, similar to the gefitinib and the alpha-tinib did. So later on, we move to our research area that we use computational first, and we screening the compound, and from the screening compound, we in the computer, and we pick up those compounds to test in the lab, and we found that it's matching very well. Another area that we did in the lab is we start from the lab first. We start from the libraries of the cyclic peptide and the libraries of nanobodies, and after that we screening by the activity assay and test it later and use computer to predict the structure of binding. So the first part I introduce here is I use the technique called the FAT display. So the thing is we have the library of nanobodies represent on the surface of the FAT display, and also the library of the cyclic peptide that represent on the FAT display. And we screening those two libraries by using the FAT display technology by coating the tyrosine kinase on the surface of the plate, and then throw the FAT library of FAT in there and watch unbound out and do propagation and do propagation again and again until we found some candidate, and we tested that candidate in the lab whether it can inhibit EGFR or not. So this is the result of the cyclic peptide libraries. So we able to identify the cyclic peptide called A1 and B1 from the laboratory from the FAT display biopanning, and this show that the A1 cyclic peptide can inhibit EGFR, but with quite high IC50 around 140 nanomolar. While B1 cyclic peptide, we got the IC50 around 4.5 nanomolar, which is similar to erlotinib as the standard, right? And we cross-check with another family of EGFR, which is the HER2. We found that the A1 can inhibit the HER2 with really low affinity at 1 nanomolar, while the B1 is not correlated to the to the HER2 at all. Is inhibit HER2 with the 321 nanomolar IC50. So in this case, we can show that we can identify the cyclic peptide named B1, and that B1 can inhibit EGFR very well and specific to EGFR and not inhibit HER2 at all. So we confirm the binding of this cyclic peptide by technique called surface plasmon resonance and comparing with erlotinib. In this case, we can show that B1 cyclic peptide have the KD around 17 micromolar, which is a little bit 2 times lower than erlotinib, which is got around 30 micromolar. Later on, we do the alanine scanning of the cyclic peptide B1. So we change the name called NP1 for that B1 peptide, and we do alanine scanning to change alanine from one by one until the last one. We found that after we change that amino acid to alanine, we found that NP13, NP14, and NP15 lost the activity of inhibit EGFR, indicate that this cyclic peptide using residue number 3, 4, 5, which is the serine, histidine, and glycine to binding with our EGFR target. So we do, and our result from the docking result show exactly the same way, that they use the residue 3, 4, 5, which is the serine, histidine, and glycine to bind on the target of to form the hydrogen bonding and bind on the target of surface of tyrosine kinase of EGFR. Later on, we try to check the activity of this cyclic peptide in the cancer cell. Unfortunately, we found that those two peptides cannot inhibit the cancer cell at all, because these two peptides cannot penetrate the membrane into the cell. However, after we adding the penetrating sequence, which are the R9 sequence in there, the B1 peptide with the R9 sequence in there can inhibit the cancer cell, which is a little bit 10 times higher than alpha-tinib. That's not too bad at the beginning point. So we got the we can show that after we add the arginine 9 in the B1 sequence, this cyclic peptide can go inside the cell and can inhibit the cancer cell, both A431 and A549. A441 is epidermal cancer cell, and A541 is lung cancer cell. So for the last part, I would like to present today is about the nanobodies. So nanobodies is a small molecule antibodies that we extract from the camel, and we make the libraries based on this variable region of the camel and put in the flat display, which we get this library from Professor Wanpenchaikampa from Mahidol University. And again, we do similar panning as the cyclic peptide we did. So in this way, we can identify that at least 3 or 4 of them, clone number 10, 35, 36, and 55 can bind with our EGFR stronger than DSA from the ELISA assay. And after that, when we check the inhibit enzyme activities, we found that 10, 18, 35, and 36 can inhibit EGFR very well. After that, we do enzyme assay. We found that clone number 18 can inhibit EGFR really, really low, IC50, and 35 is around 30 nanomolar, and 36 around 0.8 nanomolar. So it's pretty good. It seems to be much better than small molecule inhibitors in this case. And from what we see in enzyme is seems to be too good to be true. So we confirm that one with the cell line again. So it turned out that those 35 and 36 can inhibit cell line, the A549, the lung cancer cell in the nanomolar range, which is the 10,000 times better than the small molecule drug that we show here is in the micromolar range, but our nanobodies is in nanomolar range. So we confirm that it really inhibits EGFR by decrease the phosphorylation of the tyrosine 1173, as I showed before. And we do the docking study to confirm that it's possible to binding on the pocket site and block the active site of EGFR and seem to be very, very specific. And this one we did the epitope mapping to identify the binding site, and everything matching very well with our result. So that's all of my study. That's one example of the protein on the tyrosine kinase of EGFR. However, in our labs, we also do another drug study like a HIV reverse transcriptase, COVID-19 protease, the falciparum adenosine deaminase, and Leishmania triphosphate isomerase. Those are 5 compounds that we work mainly in the lab on the anti-cancer, antiviral, anti-malaria, and anti-Leishmania. So all the work has been done in Kasetsart University and collaboration with the University of Wien, University of the Nottingham, National Institute of Basic Biology from Professor Kato, and got the RTJ grant for PhD student. and also grant most of them from Kasetsart University. And this is the list of my students who work mainly in this study. Okay, thanks a lot for your attention.
Okay, thank you very much, Professor Dr. Kiattawee Choowongkomon, for your very interesting presentation and very powerful technology. So now we have time about 5 minutes for question and answer. So is there any question from the audience? You can press the mic and ask directly your question.
May I
Okay, please.
ask one questions? This is Yamada from Yamaguchi University.
Hi.
Thank you for a very interesting talk. And my question is docking screening. Actually, docking, I tried a very long time ago. So that is not so easy. Is there any good, how to say, programmed the procedure?
Yeah, I use I'm self-learning docking too, and when we try to do docking, we found that it's not successful on all cases. So in our cases, we usually depend on the we call the self-docking. We depend on the crystal structure that bind with the drug first, and we try to set the protocol that we can mimic the crystal structure. If the protocol can mimic crystal structure very well, that protocol seems to be reliable, and we can screening the drug after that. All right. So I do a lot of the proteins docking too. I can guarantee that not all cases is successful. You're right. But for my case, for the EGFR is working well. For the HIV reverse transcriptase is working well also. And also the COVID-19 docking is also looking good. We can do that. But another case we cannot docking too. So depend on case by case. That's what I can see.
I see. So you may have the very good idea, I think. Yeah, thank you very much.
Okay, thank you.
Okay, thank you. Is there any question from the audience? We still have time, maybe for 3 minutes.
Okay, if no questions, I have related questions.
Okay,
and, EGF is membrane-bound proteins, yes? So resolution is not so high.
Yeah.
So how much the resolutions of the crystals is needed to?
Or even though EGFR is the membrane-bound receptor, but somebody can cut the tyrosine kinase domain out and crystallize only the tyrosine kinase domain. So the crystal for that one is we got really and other people got really low resolution around 2.5 angstrom. So that should be enough. So you
that's quite good crystal
2.8 is enough. Yeah, 2.8 is enough for docking.
Okay, thank you. Thank you.
We still have 2 minutes. Is there any more question from the audience? Okay, if no more question, thank you very much, Professor Dr.
Thank you.
Choowongkomon. Okay, I would like to move for the next speaker. May I invite Professor Dr. Toshiharu Yaguchi from Yamaguchi University to present in the title of Engineering of Periplasmic Metabolism as a Solution for Improvement of Biotransformation Efficiency. So, Professor, please start when you are ready.
Can you hear? Can you see my slides?
Yes, Sensei, I can see.
Okay. Let me start my talk. So first of all, I like to thank the organizers to open such a nice symposium today, and thank also for letting me talk today here. So I'm going to start my talk, but yeah, it's still a bit confusing story. I think I don't like, but I think I must confuse the all of audience to understand my talk. So anyway, so please be patient with my talk for a while. Okay, I have to introduce some background about my talk today. The first one is a periplasm. Oh. Yes, first one is a periplasm. I hope all of audience have already heard the periplasm before. It's a space between the outer surface of the cytoplasmic membrane and the inner surface of the outer membrane is a gram-negative bacteria. So gram-negative bacteria has two membrane. This is a cytoplasmic membrane. Then this is the inside, the cytoplasm of the cell. Then this is a cytoplasmic membrane, and this is the outer membrane. So this side is a extracellular milieu environment. So this is outer membrane. So the periplasm is a space between two membranes. Yeah. And the important thing is, cytoplasmic membrane is a very impermeable. Yeah, it's impermeable. But the outer membrane is relatively permeable because the outer membrane has a poring protein, a kind of gate, kind of transporter to a wide variety of small molecule compounds. So that's a important feature of the periplasm. It's the environment in the periplasm is relatively similar to extracellular environment, I think. So if you consider the metabolism in the periplasm, it's another interesting feature, I think. So because the metabolism in the cytoplasmic is, how do I say, kind of complicated. It's like public transportation in Tokyo. The subway wave in Tokyo, it looks like a kind of wave in the cytoplasmic metabolism. But in the periplasm, very few metabolism, very few reaction occur in the periplasm. So that's why low competition between the cytoplasmic metabolism and periplasmic metabolism. That's another important feature. And also, of course, the cytoplasmic membrane is impermeable. So the periplasmic metabolism is isolated from the cytoplasmic metabolism. Yeah, and relatively easy transport of compounds from extracellular and to extracellular. That's another important feature in the periplasmic metabolism. So that's why, because periplasm has several features if you compare in the metabolism in the cytoplasm, that's why I interested in the metabolism in the periplasm. And another topic of my talk is acetic acid bacteria. This is bacteria is famous for acetic acid fermentation or vinegar production. Vinegar production, acetic acid fermentation is very simple reaction, just oxidize of ethanol to acetic acid via acetaldehyde as intermediate. Very, very easy reaction. But important thing is that this reaction occur at the periplasm. Yeah. So this is a feature of acetic acid bacteria, and not only ethanol, but also this is the ethanol. Not only ethanol, but this bacteria has another oxidation system in the periplasm, like glucose, like this, or sorbitol. In case of sorbitol, the end product is a 2-ketogluconate. It's a kind of yeah, raw material for vitamin C production. So this is a very important reaction in the vitamin C production industry. So I'm going to talk about kinase oxidation today. Yeah, this is the membrane-bound kinase dehydrogenase to oxidize kinase. Then the production produce 3-dehydrocyclimate. Yeah. So yes. Yeah, that's why. Yeah, that's the background of my talk today. One is what is a periplasm? What is a periplasm? What is periplasmic metabolism? I think it doesn't need how do I say? Easy to take the raw material from extracellular because outer membrane is less permeable. Outer membrane is relatively permeable. Yeah, then save energy and time to take transport across the cytoplasmic membrane. Yeah, this is expected application, saving energy and time. So today I'm going to talk extra subcellular localization of one enzyme and its modification and application with oxidation system of acetic acid bacteria. Okay, so we examined kinase oxidation system in Gluconobacter species. So this microorganism, Gluconobacter species, has kinase oxidation system naturally. So naturally, this organism possesses this kinase oxidation system in the periplasmic metabolism. I will show you later on, but this microorganism accumulates dehydrokinase in a very high yield, like nearly 100%. But that's it. So I mean, no further metabolism from dehydrokinase. This is the end product. So that's why we thought we can expand the periplasmic metabolism to produce other compound like dehydrocyclimate or protocatechuate. So if we can combine this oxidation reaction with other metabolic pathway, we can produce such compound in the periplasm. So periplasmic metabolic engineering is important because, as I said before, periplasm is very useful to transport across the outer membrane. So that's why we try to express these enzymes in the periplasm. And another thing is this molecule to help the transport of the such compound across the outer membrane. So the first story is dehydrokinase dehydratase. It's a dehydration reaction. So there are two types enzymes, type 2 DHQS and type 1 DHQS. So type 2 DHQS RQ is can be found in the widely in acetic acid bacteria. So this Gluconobacter also possess this enzyme, but the optimal pH for catalysis is alkaline. That's why it's not suitable in this experiment. So on the other hand, type 1 DHQS, ROD, this show optimum pH at pH 6.5, but it's very limited in the organism. Only 1 or 2 species of acetic acid bacteria has this enzyme. So Gluconobacter species does not have this enzyme. So that's why we clone this gene and to try to express try to express this enzyme in Gluconobacter species. So, but a problem is location. So wild type ROD is cytoplasmic enzyme. So this is a cartoon for to explain the protein secretion. This is a membrane, and this is a site of protein synthesis. If you want to transport your protein to the periplasm, you have to make this. You need to translocate your enzyme across the cytoplasmic membrane. So because ROD, a natural wild type ROD does not have signal sequence, therefore the location is cytoplasm. So we put a signal sequence at the N-terminal portion of the ROD to make a sec-ROD derivative. So there is another type of secretion system in bacteria. This is a TAT system. It's a different system from the sec type translocation system as shown before. This system, even this precursor protein has a signal sequence at the N-terminal part, but this precursor protein holds into the active enzyme in the cytoplasm. After that, translocated to the periplasm after holding is completed. So actually, we tried both type of signal sequence. One is a sec signal sequence, and another is a TAT signal sequence. So unfortunately, the sec system does not work in our experiment. I'm not sure the reason why, but anyway, I'll explain about the TAT story. So this is another enzyme in the terminal part of the acetic acid bacteria enzyme. So you can see the twin arginine motif here, RR. That is a name of TAT, twin arginine translocation system. And here is a cleavage site of the original enzyme. And we left them amino acid sequence of mature part and connected with linker sequence. And finally connect the ROD sequence itself by using genetic engineering to make such fusion protein. And this gene was introduced into the Gluconobacter species. So this is a result to produce the hydroxychimate from kinnate. So you can see the 3 lines, red, blue, and green. So green is just Gluconobacter possess vector plasmid. So it's like a wild type situation. So you can see the accumulation of dehydrokinase. This is just oxidation of kinnate. And you can see TAT strain. So this shows a nice production of dehydroxychimate. It's an end product of this experimental system. So no production with vector control, but gradually weak production can be seen with wild type. This wild type means overexpression of wild type enzyme. So this is my conclusion. Even if you overproduce wild type enzyme, the production would be slow because the location is different from the site of oxidation and site of this reaction. But if you relocate this enzyme to the periplasm, the production rate is fast. That's my conclusion in my first part of my talk. Okay, we like to expand more this reaction to another reaction to produce protocatechuate. We actually constructed a strain to produce this compound. So next, we like to combine this enzyme, dehydroshikimate dehydratase, with a previously constructed strain. Yeah, there are two types of this enzyme. The one is a cytoplasmic DHSS, dehydroshikimate dehydratase, from Corynebacterium QSUB. It's a cytoplasmic enzyme, but this enzyme, periplasmic, well, there is a periplasmic, this enzyme. It's a kind of surprise, but yeah, Acinetobacter species possess periplasmic DHSS. This enzyme is translocated by a sec translocation system. But anyhow, we overexpress this enzyme in our constructed Gluconobacter species. And another point is outer membrane protein. This may be help to translocate kinase into the periplasm. But unfortunately, we failed to construct Gluconobacter Gluconobacter strain that possess both enzymes. I don't know the reason, but anyhow, we didn't get such a recombinant strain. But we produced the strain that possess QC. It's the name of the dehydroshikimate dehydratase, and also QC and QX, outer membrane transporter. So that's why we got this strain. So therefore, we try to mix this strain and the strain previously constructed. If that work, the co-culture may produce protocatechuate from kinase by shattering the compound like this. So we try the co-cultivation. The one is a TAT-RD strain, and another is a QCX. We thought this is the best. This is most highly produced protocatechuate we thought before starting this experiment. So then these are just control combination. So vector strain instead of TAT-RD. In this case, vector strain instead of QCX. So just compare this strain with such a control strain. So this combination, number three. Number three is loss of QX. So we can evaluate the function of QX by comparison between number 3 and number 4. So these are results of the control strain. So this look like just a wild type situation. Vector control and QC strain. So therefore, just they produce DHQ, the product of kinase oxidation. And this combination is combination of vector control and TAT-RD. This is just like previously constructed strain. They produce efficiently the hydroskimate. No production of protocatechuate. So this is, as I said, that this is the best combination, TAT-RD and PCX. You can see PCA production nicely, nearly 100% yield. You can see PCA, protocatechuate. Importantly, no accumulation of intermediate species, the hydroskimate. But if you eliminate QX, no expression QX, in that case, you can see the accumulation of intermediate dehydroskimate. This data suggests QX involved in maybe kinase translocation, but also dehydroskimate translocation. So this is a summary of my second part, but I like to skip this slide. And finally, yeah, this is just I think the proof, a kind of proof of concept experiment. But periplasmic metabolic engineering is may help to increase the efficiency of the biotransformation experiment, biotransformation things. So, and finally, I'd like to thank my students, Nakamura-kun and Nagaki-kun. So particularly Nagaki-kun is our joint degree program student. So I'd like to thank his much effort to help this research project. Thank you for your kind attention. That's all my talk. Thank you.
Okay, thank you very much, Professor Dr. Toshiharu Yaguchi, for your nice and interesting presentation. So now we have time for about 5 minutes. So is there any question from the audience? Is there any questions? Or you can type on the chat board and I can read for you. Okay, Yaguchi-sensei, I have one question about the expression system in the acetic acid bacteria. So is there any previous report about the protein expression on the, the translocation on the periplasmic membrane? Is this the, first, or is this, some people already try some signal peptide to translocate the protein expression?
Actually, not the first. I think the second experiment, but it's the previous work is a little bit complicated because they use the secretory protein, originally secretory protein, but fused E. coli signal peptide. How do I say it? Exchange the original signal sequence with E. coli signal peptide and expressed in acetic acid gluconobacter species. Yeah, I'm not sure why they do such experiment, but yeah, the previous report did such experiment. So that's why this is not fast.
I see. But I think the idea is quite interesting about to express the enzymes on the polyplasm to continue the reaction and make it fast for the production. So is there any more question from the other audience? Please. Okay, if no more question, thank you very much, Professor Dr. Toshio Yakuchi.
Thank you.
So next, I will move to the next speaker. So may I invite Dr. Panaya Kotchaplai from Chulalongkorn University to present in the title of Performance of Phenol-Adapted Microbes on Palm Oil Meal Effluents or POME treatments. So Dr. Panaya, if you are ready, please start. Okay, I can see your slide.
Okay, so thank you very much. Good afternoon, everyone. My name is Panaya Kotchaplai, a researcher from the Institute of Biotechnology and Genetic Engineering, Chulalongkorn University. So for today, I'm going to talk about a part of my research that was conducted last year along with the unstable COVID situation. So the topic is the performance of phenol-adapted microbes on palm oil meal. In this presentation, I will call it POME treatment. Okay, so the world population is increasing and it is expected to reach 2.8 billion in 2050. And along with the increasing world's population, it means that the increasing demand of the food, including the vegetable oil. So it has been reported that the world consumption in the past 2 decades for vegetable oil increased by 2-fold. And this leads to the concern on that how can we produce the sustainable vegetable oil to feed the growing world population without disturb the natural environment. So according to the World Economic Forum, so palm oil is one of the proposed solutions for this problem, since palm oil plantation require a less cultivation area compared to other oil crops. So from this slide, you can see that palm oil contributes to 1/3 of the global vegetable oil, while it requires only 6% of the total oil crop cultivation area. So when we take a look at Thailand is among the largest palm oil producer in the world, after only Indonesia and Malaysia. And palm oil production in Thailand contribute to approximately 3 to 4% of total global palm oil production, which is about, so it's in 2019 it was about 2.6 million tons. And for the palm oil milling activity or the process to extract oil from the oil palm, in Thailand so far we have about 150 palm oil mill, which with annual production capacity of 2.8 million tons. However, together with the increasing palm oil production activities, it leads to the increasing concern on regarding the environmental impact. So when you take a look at the palm oil milling process here, from these 3 steps, it can generate the wastewater, or which will be called palm oil mill effluent or POME. For 1 tons of crude palm oil production, it can generate 2.5 tons of POMEs. And so what does POME look like? So when you take a look at the picture here, POMEs is a oily wastewater. It's very thick, it's colloidal, the color is brownish or yellow, and it contains a lot of high organic content. So due to the presence of the fat and grease, suspended solid and lignocellulosic biomass. And the typical palm oil POME characteristic is in the acidic nature. Its pH can be ranged from 3 to 5 with high salt as well. Okay, and besides the concern regarding the wastewater generation, the anaerobic degradation process of POME also generate biogas, which also concern as the contributor to the global warming. Okay, so since POME is characterized by the high organic acid content, the carbohydrate, mineral, and proteins, so it makes this wastewater as a source for further growth of biomass. So these are 2 POME management that has been proposed and practiced recently. So this is the cover lagoon and the continuous stir tank reactor, which generate biogas. So the biogas, or particularly methane that obtained from this step, will be used to generate electricity inside the palm oil mill. Okay, beside the attempt to reduce the release of greenhouse gas, so on the right-hand side is the ideal POME treatment to reduce the release, to reduce the disposal of wastewater. So from this, so once POME, raw POME is treated by the anaerobic digestion to generate biogas and anaerobic tank. So typically the discharge from here will be released to the environment. So, but in this scheme, they try to, they propose that the use of this discharge to input as an input for further purification process to further create the clear water enough for use for boiling process. But that was the ideal POME treatment in most of the large palm oil mill. But typically in the much smaller or medium, the small or medium scale palm oil mill, typically like commonly found in Thailand, they typically use the open lagoon system or the series of stabilizing ponds that consists of oil trapping pond or oil pit to recover back the residual oil, the cooling pond to reduce the temperature of the raw pond, the anaerobic pond to reduce the COD coupled with followed by the aeration of aerobic pond before polishing and discharge. Okay, and this is normally it looks like it's just a large pond connected together, so it will flow into the serially. So when you take a look at is the retention time of each step, you can see that it's quite a long time, like from 6 days to 7 days to 60 days, 20 days in each step. Okay, and even though it is a low cost and simple in terms of operation, it is a limit. The limitation of the open pond is that the land and time consuming, and the improper management of POME, especially by using the local stabilization pond, always lead to the environmental impact. So from this three figure, it is like, I believe it's 5 years ago in Krabi province, Thailand, that the sudden rainfall result in the sudden increase of the amount in the stabilization pond, and this POME was released to the nearby water body and affect the daily life of people surrounding that area. Since the biological treatment is low cost and easy to operate, the biological based pond system seem to be the most suitable. But however, since it is also time consuming, large area required due to high retention time in each step, and also the low efficiency. So these three are still the challenge for biological treatment of POME. So it has been reported that the parameter that result in the low POME treatment efficiency, the number one is the chemical inhibition. So since, as I mentioned earlier, that POME also contain a high amount of lignocellulosic biomass, which consists of cellulose, hemicellulose, and lignin. So lignin is a complex structure of aromatic compounds. So upon the degradation, it will release a heterogeneity of the serotype of the aromatics. And this can be toxic to bacterial cells and inhibit their activity. And beside the phenolic compound is also the volatile fatty acid and heavy metals that can inhibit bacterial activity. And the residual oil in POME can also cause the oil encapsulation of bacteria. So prevent bacterial cell to further degrade the other compound within POME water in POME. And one challenge of the POME treatment efficiency is the high variation in POME characteristics. Since the characteristic of POME, which mean by the content of organic compound, the content of phenolic compound, depends on various factors, including the season, of milling process, the processing technique, the type and age of palm oil. So these parameters altogether can result in the low POME treatment efficiency. And this also makes the final discharge of POME exceeds the regulation limit of Thai effluent regulation. So since phenolic acid is one of the most concerned compound in POME, so several studies try to deal with this compound. So this is the study by Janto. This is study from the research group of Associate Professor Dr. Orama Sittinun from Prince of Songkla University. They try to use the Bacillus thermorevolans A2 or the phenol degrading bacteria to reduce the amount of phenolic acid in POME. So since even though gallic acid is the most presented phenolic acid in POME, it can be reduced to acceptable level. However, when you take a look at phenol, even though it is aerobic or anaerobic reaction, phenol acid high toxicity is still become the major recalcitrant compound exists in POME, even though after the final step. So accordingly, I am interested in phenol removal. So, and to my concerns, if we allow bacterial cell to adapt to phenol, which will result in their higher tolerance, can bacterial cell adaptation to phenol improve the POME treatment efficiency? So beside the indigenous consortium obtained from the POME salad, I'm also interested in Bacillus groups since they are commonly found in POME. So these two isolate Bacillus, W1 and CU2-1 are the two strains I'm working with. So these two strains are the lignin degrading bacteria that can degrade lignin and accumulate a certain types of phenolic compound. Thus, so it's quite tolerate to the certain level of phenolic acid. So when we take a look at the COD removal or chemical oxygen demand removal in POME by using these three microbes, the W1, Bacillus W1, the Bacillus CU2-1, and the consortium, we found that in the first cycle, which we use 5% POME together with 100 milligram per liter of phenol, the removal efficiency of these three bacteria are quite similar, which is about 50% removal. But when we further allow the adaptation or subculture these three group of bacteria into the new POME supplemented with phenol, we found that even though the W1 and CU2-1 are the lignin degrading bacteria that can tolerate the high amount of phenolic acid, the COD removal efficiency decrease, while the COD removal by the Phenol-Adapted consortium improve. So for the consortium from 50% to 75% removal. One cell were adapted to phenol for 3 cycles. Then both non-adapted and phenol-adapted microorganisms were then tested for their removal, COD removal in comparison as shown in this slide. You can see that once adapt for the lignin degrading bacteria, Bacillus W1 and CU2-1. So once adapted to bacteria, their COD removal efficiency decreased. On the other hand, the COD removal by the phenol-adapted consortium slightly increased compared to the non-adapted consortium. So from the phenol-adapted consortium, I did isolate 5 microbial stain based on the their ability to produce esterase and lipase by using the tributylene and rhodamine B plate. And interestingly, let's focus on the lignin degrading bacteria first. So the result was like the previous experiment, that the presence of phenol reduced COD removal, which correlate with the phenolic acid removal, since these two strains can accumulate a certain species of phenolic acid. However, when we take a look at the consortium, so even though the consortium can remove COD by 70 to 77%, the maximum COD removal by the isolated strain was only 21%. However, but once phenol was added to the medium, this phenol addition increased bacterial removal efficiency. It can be from 7% to up to 35%. And when we look at the phenolic acid removal, it seems like it's strain dependent and not correlated with the COD removal. So it's a bit quite complicated, as you can see that to be more understand about this observation, so it is needed to identify the metabolite, particularly the phenolic compound that presents in this experiment. However, considering the practicability of the phenol-adapted microbe, so the consortium was selected for further study. Okay, and unfortunately, during COVID-19 situation, that most of our research facility was shut down. So I have to halt the phenol adaptation experiment, and once resume the research activity, I found that I found a major change in bacterial community as well as the activity. So we have to start our experiment all over again. So in this time, we increased the initial concentration of POME from 5% to 25%, which give the initial COD of about 12,500 to 15,000 milligram per liter. And you can see that this is just the comparison between the COD removal in 25% POME and COD removal in 25% POME added with 100 milligram per liter phenol. So you can see comparable COD removal within 3 days. So the maximum removal was about 75%. However, when we take a look at the phenolic acid removal, we can see that the presence of phenol, the consortium with 100 milligram per liter phenol show slightly around 22% higher phenolic acid removal. So from this, it can be seen that it is appear that the addition of low amount of phenol can stimulate the phenol degrading activity. However, this not affect the overall COD removal. This probably due to the low amount of phenolic acid in POME comparison to other organic content, for example, fatty acid or carbohydrate. So for phenol adaptation experiment, the consortium was allowed to adapt to the increasing concentration of phenol from 100 to 250 milligram per liter. And when we take a look at the light pink marker, so for both COD removal and phenolic acid removal, the maximum removal was achieved at 67 to 70% within 3 days. Okay, and when the treatment was prolonged, only little higher removal was observed. But once this adapted cell was challenged with much higher phenol concentration, for example, in this experiment, I used 500 milligram per liter, which is 2 times higher concentration of phenol. You can take a look at the bright red here. So we found the 3-day log phase in COD removal. And for when we take a look at the phenolic acid removal, we can see that only 28% of phenol was removed before and remained stable before it starts to rebound again after prolonged treatment. But once this the previous consortium was adapted to 5 milligram per liter phenol, the consortium then able to remove COD and orthophenolic acid compound in similar manner to those in lower phenol concentration. As you can see, it showed a comparable pattern with the light graph, which showed that consortium degradation, COD removal by consortium in 25% POME together with 250 milligram per liter of phenol. Okay. So this is the experiment to see that if there any difference in COD removal between the normal and phenol, the consortium adapted to high concentration of phenol in the phenol-free POME. So it's like to see the stability of their activity. So we see the comparable COD removal of which around 74 to 78%. However, for the phenol-adapted consortium, we still see the around 20% higher phenolic acid removal. So from this, it can be concluded that effect of phenol adaptation is much more pronounced in phenolic acid removal, while a less challenge with a much higher phenolic acid concentration. The COD removal efficiency between non-adapted and phenol-adapted consortium are quite comparable. And unlike the adaptation in a single strain, which can be a result from change in the physiological state, gene expression, or even change in their genome, the adaptation in a microbial consortium is quite complicated, since it can be attributed to even the distribution and the activity of each species in the consortium. So it seems like that this is still an initial step for further develop a practical microbial consortium for POME treatment, and many aspects are still need to be explored. Okay, so finally, I would like to thank Ueda Overseas Research Grant from awarded by Kiattawee and Environment Foundation Japan for financial support, the research unit in bioconversion and bioseparation for value-added chemical production, and also Professor Alisa Wangnai from the Biocatalyst and Environmental Biotechnology Research Unit for the facility support. Thank you very much, and that will be all of my presentation today.
Okay, thank you very much, Dr. Panaya Kotchaplai, for your interesting presentation. We have time about 5 minutes for the question and answer. So is there any question from the audience, please? Okay, Imai-sensei, please.
Thank you very much for your good presentation, excellent presentation. I have one question about the adaptation. Can you hear me?
Yes.
Yeah. The adaptation, it's the adaptation itself is the more exceed or not? The more for example, the COD removal rate is the more longer time adaptation is the more higher the efficiency you can get.
You mean the
adaptation period? The more longer period is the more higher adaptation or not?
It depends. We have to see, because it's not always the case that the more adaptation will result in the more efficiency.
Mm-hmm.
So if we want to know this, we have to conduct the experiment. So it cannot be concluded by.
I see. And in the pond of the palm oil the treatment pond, that pond is have so many high tolerance of the phenol tolerance bacteria or not? Maybe you can find.
So in actually the most of the phenol tolerance was isolated from POME sludge, so which settled down in the pond. So what we find the consortium in this experiment was also obtained from the sludge.
I see. So the result of your experiment or the research is the consortium itself. Consortium is more higher efficiency, right?
Yes. Yes, because
so, yeah.
Even though the isolated strain show the process the key enzyme, for example, lipase or esterase, it singles the COD removal efficiency of each strain much lower in comparison to the consortium. So we think that it's need the co-culture or the co-activity between each strain so that the COD can be removed effectively.
I see. So I can understand it is the biosynthesis. It's a biosynthesis of the many bacteria.
Yes.
I see. Thank you very much.
I think now the time is up. Please, thank you very much, Dr. Panaya. So I will move to the next speaker. May I invite Associate Professor Dr. Patchareeya Boonkorkaew from Kasetsart University to present in the title of Dendrobium Orchid Industry in Thailand, Research for Resilient and Sustainable Production. So if you are ready, please start.
Just a minute. I think I have some problem. I will share again. Okay. Did you see my PowerPoint?
Yes, actually. Oh, okay. Now it's okay. Yes.
Okay. Thank you for introducing me, and good afternoon, everyone. Minasan konnichiwa. I'm going to present my research entitled Dendrobium Orchid Industry in Thailand, Research for Resilient and Sustainable Production. Cultivation of orchid in Thailand started in 1916 and until 1966 with export to European countries. Thailand has been the world's largest producer and exporter from 1979 to present, especially the production of Dendrobium orchid cut flower. The main production factor that you know are genetic or varieties, environmental and cultural practice or management. The advantage of orchid industry of Thailand are many varieties of orchid cut flower that you see in this picture, not only in Dendrobium orchids, but also in Mochara or Aranda or Vanda. And also in Thailand has a suitable environment for the orchid growing. Dendrobium sonia earskun and Dendrobium khao are the most popular orchid cut flower cultivar for exporting. They are fast growing, floriferous, bright color, and have a long waist life. Dendrobium orchid cut flower production typically involve plant grown on coconut hut and less branch under 50 and 60% shade net. That is very simple construction of greenhouse in Thailand. You know, when my friend, they came from the Netherlands, they said, oh, Thailand is the largest in the world to export orchid to around the world. But why the construction of greenhouse is very simple because of the table of environment for growing orchid cut flower in Thailand. Environmental factors for the orchid requirement are approximately 25 to 35 degrees Celsius in the daytime and in the nighttime more than 18 degrees Celsius, with relative humidity about 60 to 85%, and with light intensity around 800 micromole per square meter per second, and rainfall annually 1,200 millimeter. That is where is similar central Thailand environment. So the production area is mainly in the central Thailand, especially Nakhon Pathom, Samut Sakhon, Ratchaburi, Bangkok, and Nonthaburi, where the Chao Phraya River and the Tachi River are the water sources for cultivation. Orchid grower can produce orchids at a competitive cost with a suitable climate and more than 40 years of orchid growing experience. That's why we become the largest producer and exporter in the world. However, currently, they are encountering drought and environmental affecting due to climate change. Orchid growers face climate variability that affect growth, flowering of the orchid and subsequent production. We try to solve this problem by developing a greenhouse for coping with temperature and precipitation fluctuation. So we study pattern of photosynthesis, environmental factor, and also we try to develop the greenhouse. So that's why we have to study in the basic research in the pattern of photosynthesis and also environmental factor in the different greenhouse.
Excuse me, Dr. Patchareeya, can you speak more a little bit loudly? Because
did you hear me?
Actually, I can hear you, but some other maybe can hear very small voice.
I'm not sure because of microphone or not. But and now you hear me.
But
I will try to check. Now you hear me? Excuse me.
Right.
Now it's okay or not?
Right. Stop. ช่วยเช็คได้ไหมครับ
It's better. Okay.
Okay.
I take it off my ear headphone. Okay.
Thank you very much. Please continue.
Okay. So we found that Dendrobium orchid is a campant. So stomata open in the nighttime and least maximum about 2 to 6 a.m. So that we can supply CO2 800 to 1,200 ppm for increasing photosynthetic efficiency. And also the optimum temperature is 22 to 29 degrees Celsius. Relative humidity around 60 to 90%. And also the light saturation point is around 200 micromole per square meter per second. And we can introduce the natural air for plastic greenhouse is an alternative net shade greenhouse for the farmer because they can control the temperature and fluctuation of precipitation. Moreover, we study water requirement and water salinity effect for coping with the water salinity because of the Royal Irrigation Department cannot release water into the Chao Phraya River due to the drought crisis. Low water level in the Chao Phraya River is very low. So in this case, the seawater will be introduced to fresh water. You can see this is the Patchareeya River and the Tachi River. Both river is the water source for orchid production in the central Thailand, and is not far from the Gulf of Thailand, just only about 60 kilometers. So it's very easy to see water intrusion. So Thailand, in 2014, Thailand fed to the sea water into to fresh water. So Thailand suffered the worst water salinity level in more than a century. The salt concentration of water in the Tachi River was higher, let's say around 10,000 to 16,000 microsiemens per centimeter. The estimate orchid cultivation area affected by this water salinity was approximately 1,600 hectares, or 50% of production area in 2015. Since orchid growers do not know much about the effect of salinity, they are afraid to using saline water to water orchid plants, and they let orchid die. And some growers bought fresh water to water. That is effect cost is higher. And some orchid growers, they try to use some water that is effect from the sea intrusion. So water in the reservoir is become saline water, and then orchid will become yellowing and wilting. We investigate how Dendrobium orchid is developing state, tolerates saline water for 1 month, 2 months, 3 months. And when water with a high salinity water, this is a level of water that we measure by EC meter. It's like when water with a high salinity is mean leaf and stem show yellowing and wilting, especially in the young plant state, and some mature and flowering state is still okay. But if high, I mean 8,000 microsiemens per centimeter, the orchid is become yellowing and wilting and die finally. At 8,000 microsiemens per centimeter, orchid no flower. But in the level of control, compared with the 2,000 and 4,000 microsiemens per centimeter, the orchid have some flower. But the quality of flower in 4,000 microsiemens per centimeter is quite different. It means the quality is will be reduced. The size of flower is become smaller, and number of flower is reduced. When measuring the net photosynthesis rate of Dendrobium by Lycor 6400XT, this portable photosynthesis system to measure the net photosynthesis in 24 hours from 8 a.m. to 8 a.m. The result demonstrates that net photosynthesis in every state decreased with the water salinity increase, especially during maximum stomata opening at 4 a.m. But if you see in the EC about 2,000 is maybe quite higher than control, but it's not different. Also, number of roots also decrease with water salinity increase that you see from 1 month and 2 months. Percentage of new shoot and new inflorescence after treatment for 2 months were approximately 60 to 100%, respectively in 2,000 microsiemens per centimeter. Flower quality was high and not different after treatment when after treatment 1,000, 2,000, and 3,000 when compared with control. And also this is when we to water for 2 months, except for 2,000 microsiemens per centimeter, the plant is quite low quality. We also find out the water requirement of Dendrobium in each development. In this, we can see the grower can use this research to save the water in the reservoir during the dry season, because nowadays, from the past time, I mean 40 years ago up to now, the farmer, they use the water by sprinkler, and sometimes they open the sprinkler, take a long time, about I think about 15 minutes, and they use water, a lot of water that is not necessary for the plant requirement. So that's why we have to do some experiment about the water requirement in orchid. And let's say when we produce Dendrobium orchids, is take time about 5 years from the young plant to flowering plant. In this case, we found that pilot state require 10 milliliter per plant a day, and for mature plant, about 50 millimeter per plant, and for flowering state is around 60 millimeter. That is mean when you look at here is a quite low water requirement for Dendrobium orchid cut flower. We conclude that Dendrobium's salinity tolerant EC should be less than 1,000 microsiemens per centimeter in case of pilot state or young plant state. And in case of mature and flowering state, we say EC should be less than 3,000 microsiemens per centimeter. And farmer can use that kind of water no longer than 2 months continuous. The grower can use their water in the reservoir when the water become saline water. It's like let's say during dry season in Thailand, start from November to February, or sometime to March, or sometime to April, depend on the rainfall also. So in this case, in this year, the rain is become faster than last year. So the Tachi River nowadays, the EC in the Choowongkomon River and Tachi River is quite normal. So depend on the precipitation also, and also depend on the climate in each year. So when we have some research, this kind research and introduce to the farmer, the grower can use their water in the reservoir using an EC meter to measure the water. If the EC level is over the tolerant level, we recommend they dilute by fresh water. We also transfer the knowledge and solution to orchid grower. Currently, they are confident to use saline water. I mean, the fresh water in their reservoir become saline water during dry season. They are confident to use their water from the reservoir, canal, or the river using the EC meter before use. And all of our selected publication from 2002 to 2020 that I present today, we have continued to do research for sustainable Dendrobium orchid production industry and maintain the largest producer and exporter of Dendrobium orchid industry in Thailand. So we have to continue our research, and sometimes we have to forecast the problem in the future also, and try to do some experiment and some research to cope with the situation that it will come to. So last but not least, I would like to thank my PhD and master student for making the effort to do the research, including my colleague and the funding organization in Thailand and in Japan. And also thank you very much for your attention. And thank you for KU and YU to organize this symposium. Thank you very much. ขอบคุณค่ะ
Thank you very much, Associate Professor Dr. Patchareeya Boonkorkaew, for your nice and interesting presentations. So we have time for 5 minutes for question and answer. So is there any question from the audience? So I have asked one question about, actually you present some result about the salinity, I mean the effect on the orchid
Yes.
flower formation. So how about other factor that is also affect the production of the orchid?
Yeah, sure, right. Because of the effect of the production, orchid production, as I said before, depend on the variety. So in Thailand, there are so many variety that is okay for us. We still have there are so many variety, but the effect that is important to produce orchid cut flower for industry. That is an environmental factor. Not only salinity, water salinity, but also in temperature and also humidity. That's I say why I have to do some basic research to find out the optimum temperature and also humidity that is suitable for orchid growth. That's we found that's about 25 to 25. This is the optimum in the daytime. But from 22 to 29 is very okay for orchid production in the daytime. And also Dendrobium orchid is the tropical plant. So we have to control in the nighttime, not more than and not less than 18 degrees Celsius. So that's why we try to develop the greenhouse for the farmer also. Because if you see in the picture, that is very simple construction for orchid greenhouse, and that's why we become the largest producer and exporter because of the low cost production. If you try to change the construction of greenhouse like a plastic greenhouse, as I say, we have to introduce the farmer to use some construction that is very easy. That is a simple to use also. So, and also light intensity, that is important for orchid growth. That's why I say about 200 micromole per square meter per second. That is we list the light saturation point of production. If you go orchid lower than 200 micromole per square meter per second, the flower production will be reduced.
Okay,
and
thank
also
you.
the quality.
Thank you very much.
Thank you.
So just one more minute. Is there any more question from others? Okay, if no more question, now the time is up. Thank you very much, Associate Professor Dr. Patchareeya.
Thank you very much. Arigatou gozaimasu. สวัสดีค่ะ
Okay, we are going to have a short coffee break for about 20 minutes. So please come back here by 2:50 Thailand times or 4:50 in Japan times. So thank you very much. Okay. Welcome back everyone to the 7th Priority University Symposium. So we are going to continue for the next session. Next speaker, may I invite Associate Professor Dr. Yuki Sako from Yamaguchi University to present in the title of Development of a Leaf Lettuce Growth Model in a Plant Factory. So if you are ready, please share your slide and start.
Thank you very much for introducing.
Okay,
Today
we can see your slide.
Thank you. Today I'd like to talk about plant factories. Recently, yield and price of vegetables have been unstable because of extreme weather events in Japan. Plant factory is a controlled environmental system to produce high-quality crops all year round without being affected by natural weather events. In a plant factory, environments are fully controlled, such as air temperature, humidity, electrical light, carbon dioxide, nutrients, and water to achieve highest productivity. And in plant factory, cultivation can be three-dimensional like this. And leafy vegetables are mainly cultivated because it can be produced in high yield and they have high market value. And light quality is most important factor in environmental control in plant factory, and it can be controlled with LED. And in addition, fertilizer management is also important in a plant factory where hydroponic is used. However, in plant factories, plants are grown with low light intensity and high nitrate concentration in nutrient solution. And as a result, nitrate concentration in plants tend to be high because plants store excess nitrates in vacuole. In addition, this condition leads problems such as increases in fertilizer cost due to excess fertilization and environmental loss from waste of nutrient solution. Therefore, fertilizer management according to growth rate is required by maintaining appropriate balance of root nitrate absorption and carbon fixation by photosynthesis. And because these depend on cultivation environment, it is necessary to determine the optimal environment for cultivation by modeling support. We have developed a model of carbon and nitrogen absorption and metabolism of plants that can estimate nitrate concentration in leaves, which is an index of excess fertilization based on the balance between absorption and metabolism of nitrate. However, in this model, the plant growth is expressed by a simple logistic equation and not a mechanical model. So therefore, in this study, we developed a mechanical growth model for leaf lettuce considering the photosynthetic performance. Our research consists two parts. First, we study an estimation method of photosynthetic rate per plant, and then we develop a leaf lettuce growth model for estimation growth rate from carbon assimilation rate. And then The model was used to estimate the growth rate under different fertilizer conditions. Leaf lettuce cultivar frill ice and L122 were used for plant materials. And we have a plant factory facility in the campus, and all experiments were conducted in the facility. And experimental conditions like this. And measurements were photosynthetic rate, fresh and dry weight, leaf area, carbon content. To measure projective leaf area, PLA, the photo of plants taken from a height of 110 centimeters above, PLA was measured. Then image was analyzed to measure PLA by using leaf area analysis software. For the measurement of total leaf area, all leaves were scanned like this by scanner. Then PLA was measured by using same software. For the measurement of photosynthetic rate of single leaves was measured by infrared gas analyzer LI 6400XT, and the photosynthetic rate of individual plant and community were measured by using assimilation chamber. Light intensity was set to 0 to 350 micromole square meter per second. Measurements were conducted at between 25 to 40 days after sowing for single leaf measurement, and 20 days to 40 days after sowing for individual plant and community measurement. This is a schematic diagram of evaluation system of the photosynthetic rate of plant by assimilation chamber. This system consists of air pump, flow meter, assimilation chamber, and carbon dioxide sensor and data logger. Air was supplied to assimilation chamber, and carbon dioxide concentration of the exhaust air is set to around 1,200 ppm by controlling carbon dioxide concentration in the supply air. The supply air and exhaust air were sent to carbon dioxide sensor to measure carbon dioxide concentration by switching three-way valves. The photosynthetic rate was calculated by using this formula. This is the formula or model of light response curve of photosynthesis, where A is photosynthetic rate and G max is maximum photosynthetic rate. K is Michaelis-Menten constant, which is a light intensity at the photosynthetic rate of half of the maximum photosynthetic rate, and RD is dark respiration rate. This shows the photosynthetic rate under per plant under individual condition, under different light intensity and different growth stage. The photosynthetic rate per plant under individual condition increases with increasing light intensity and represents light response curve like this. And photosynthetic rate increase with increasing plant growth. This shows photosynthetic rate of plant community. The the photosynthetic rate per plant measured under plant community condition. The photosynthetic rate under community condition was higher with higher light intensity, but there were no obvious different photosynthetic rate after 30 days after sowing. This shows the photosynthetic rate of single leaves. The photosynthetic rate of single leaves were higher with higher light intensity, and the photosynthetic rate was getting lower with the growth of the plant under a high light intensity. From this result, the relationships between the fresh weight and maximum photosynthetic rate and Gmax and Michel's maintenance constant K were obtained. The high correlations were obtained. Therefore, it is shown that Gmax and K can be estimated from the fresh weight using this equation. This shows the relationships between leaf area and photosynthetic rate. Blue A is the product of the total leaf area and photosynthetic rate of single leaves, and yellow B is photosynthetic rate per plant under individual condition, and green C is product of projective leaf area and photosynthetic rate of single leaves, and D is photosynthetic rate per plant under community condition. The actual photosynthetic rate per plant under individual condition and community condition were significantly different, and the estimated photosynthetic rate per plant calculated from total leaf area was overestimated than actual value. The estimated photosynthetic rate calculated from projective leaf area corresponding to well with actual photosynthetic rate under community condition. In the plant factory, light is directed from above, so not all leaves are exposed to light, and some area shaded by overlapping leaves under community condition. So from this, the estimated value of the photosynthetic rate per plant obtained from the product of PLA and photosynthetic leaf, photosynthetic rate of single leaf can apply to the growth rate model. This shows the relationships between PLA and dry weight. The high correlation was obtained, so it is shown that PLA can be estimated from dry weight using this formula. This shows the relationships between dry matter ratio and dry weight. Dry matter ratio also can be estimated from dry weight. The model components can be summarized as this. All model components are obtained from the equation of dry weight. As a result, we developed this leafletous growth model. This model calculated the time course of fresh weight based on the dry weight dividing by the dry matter ratio. And the dry weight was estimated based on the photosynthetic rate and PLA, which were calculated using this formula. And dry matter ratio was calculated using this formula. And alpha is the carbon dioxide fixation to dry ratio of carbon dioxide fixation to dry weight increase. And 2.5 was used in the previous research. This is the time course changes in carbon content in leaf lettuce leaves. Carbon content were measured by element analyzer using the measured value of carbon content. The coefficient alpha was calculated. The value was close to literature value of 2.5, so the 2.5 was applied to in the growth model. We then simulated the time course changes in fresh weight and the different light intensity. The dots show the actual value of the fresh weight of leaf lettuce grown and the different light intensity. The data calculated sequentially by applying the dry weight of 20 days after sowing as the initial value, like this. As a result, the simulated time course changes in fresh weight of leaf lettuce grown under both light intensity corresponded well with actual measurement. We then use the developed model to estimate growth rate under different fertilizer conditions. Leaf lettuce were grown in 3 fertilizer treatment, EC 0.5, 1, and 2 decimals per meter from 20 days after sowing. Leaf nitrate content and leaf photosynthetic rate, fresh weight, and dry weight were measured every 5 days. This graph shows the result of nitrate content in leaves. Nitrate content in leaves was lower with decreased EC. According to previous studies, there is positive correlation between the photosynthetic performance and nitrogen content in leaves. So this graph shows the leaf photosynthetic rate. However, there is no relationship between leaf photosynthetic rate and EC and leaf nitrate content. In the previous study, experiments were conducted in the outdoor. In the plant factory, the cultivation environment is suitable for the plants, so the photosynthetic rate could be sufficiently high under any fertilizer condition. Therefore, the leaf photosynthetic performance may not be significantly affected by the nitrate content in leaves. This graph shows the relationship between dry matter ratio and dry weight. The dry matter ratio increased with decreased in C. According to previous study, nitrogen stress lettuce has a high dry matter ratio than lettuce grown under normal condition. So it's suggesting nitrogen stress caused under lower EC also increase the dry matter ratio in this study. Based on this result, we apply the parameter of leaf photosynthetic rate and dry matter ratio, which varied among the treatment. The simulated value were well matched the measured value in the all treatment. It is indicated the estimation of growth rate under different fertilization fertilization condition required two parameters of leaf photosynthesis rate and of dry matter ratio. I'd like to conclude. From this research, we were able to develop a leaf lettuce growth model that estimates the growth rate from the carbon assimilation rate by estimating the photosynthetic rate per plant based on the dry weight at transplanting. The leaf photosynthetic parameters that can be measured non-destructively, and the projected leaf area that can be photographed with a camera during the cultivation. The model was able to evaluate change in growth rate caused by changing environmental conditions and photosynthetic performance. And using this model, it is possible to estimate the growth rate when the fertilizer management is changed, which can be applied to the optimization of environment control condition in plant factory. Thank you very much.
Okay, thank you very much, Associate Professor Dr. Yuki Sako, for your interesting presentation. So we have time about 2 minutes for question and answer. So is there any question? Professor Alisa, please.
Yes, thank you very much for your interesting talk. Since your work is regarding to the indoor plant factory, from what I heard, it's that the heat generated from the light source possibly involved in the plant growth. So I'm wondering if your growth model has accounted for heat generation from the light.
Yes, and thank you very much. The temperature is also affect growth rate. So we have to conduct the experiment under different different air temperature. We didn't conduct under this different temperature. Okay. So I can't answer that question, but it might be affect the plant growth rate and plant growth model, parameter of growth model.
All right. Okay. So I'm looking for the next stage of your work. Thank you very much.
Thank you very much.
We still have 1 minute more. Is there any more questions from the audience? Okay, if no more question, thank you very much, Associate Professor Dr. Yuki Sako.
Thank you very much.
Okay, I will move to the next speaker. May I invite Associate Professor Dr. Sasimanat Unnachak from Kasetsart University to present in the title of Regional Chimeric Vaccine Design, a platform for producing multimeric vaccine for aquaculture. So please share your slide and start the presentation. Okay, now we can see your slide.
So, konnichiwa. Good afternoon, everyone. I'm Sasimanat Unnachak from Department Biochemistry, Kasetsart University. So today is my honor to be here to talk about the life underwater. So I would like to move all of you guys to the aquacultures and how we can develop the vaccine for aquatic animals. As you see in the figures, we use Nile tilapia as a model for vaccine development. The Nile tilapia has really long times histories. It's come to Thailand in last 60 years ago as a gift from the Japans. And now the tilapia is counting as the economically fish that can be cultivated in Thailand and also export to other countries. But however, the tilapia cultivation is faced with a lot of the disease. As you see here, we culture the tilapia in different areas in Thailand, and it's found that it's faced with the infectious disease that cause mass mortality, and sometimes it's caused economically loss. So upon we doing the disease surveillance, we can conclude that the tilapia disease can develop from either the bacterial disease and also the viral disease. For example, the streptococcosis that come from the Streptococcus agaricales in tilapia in Thailand, Aromonas from Aromonas species, and also the Flavobacterium. We also found some of the viral disease that affect the tilapia aquacultures. For example, tilapia leg virus that caused the disease in the beginning of tilapia cultivations. But however, upon our focusing, we're looking for the serious disease that can cause the disease and cause a mass mortalities along the 4 to 8 months of the cultivations. So the first disease that we focus is the streptococcosis in fish. Why we focus on the streptococcosis? This disease is mentioned as a common disease that can occur in the fish worldwide, not only the tilapia. As you see in the figure, this shows the area that culture the tilapia. In the China, Indonesia is 2 major countries that produce a lot of tilapia. And these 2 countries also affected by this disease as well. And this disease caused by the Streptococcus agaricales, and we found that it has 2 major strains. It's called serotype 1A and serotype 3 that affect to the cultivations. But however, if you see from some area, they cannot classify the serotype of this disease. So this is the reason why the streptococcosis vaccine that produced by the commercialized company is not functioning. And from the food safety and security dimension from the OIE and also the FAO and WHO, they led us to prevent the antibiotic use in aquaculture or agricultural productions. And the vaccines in animals is supposed to be one of the solutions for prevent of the infectious disease. So this is the reason why we interest to develop the vaccine to be used in aquacultures. And the core technologies in the vaccine development nowadays, including in the human being, we can separate the core technologies to be 2 types. First one we call conventional vaccine development. The second one we call reverse vaccinologies. How different between them? The conventional vaccine development is use the whole organism that characterize and produce the vaccines as inactivated or killed or pathogens or live attenuated or weak pathogens. But we also have the new technologies called reverse vaccinologies that produce the vaccine from just only some part of the pathogens that we can classify them as antigen proteins. So in case of the reverse vaccinologies, we need the technologies that can characterize or identify the antigenic proteins to be produced the vaccine. So the techniques that mostly used are the proteomics analysis or whole genomics analysis. Then we need to evaluate the vaccine candidate in silico analysis pre-production as the vaccines. So in these reverse vaccinologies, they also have 2 types of the vaccine as well. One is called recombinant protein vaccine. The second one is called subunit DNA vaccine. But both of them can be used as a vaccine in animal as well. So in our technologies, we use the streptococcosis as a model. we developed the inactivated vaccine. We success to produce Strep-KU1 or formalin Q vaccines as a model. And we also produced the subunit protein vaccine as Strep-KU2, and Strep-KU4 is a chimeric DNA vaccine and protein vaccine. In case of Strep-KU3, it is a combination between Strep-KU1 formalin Q vaccine and subunit protein vaccine. So in case of Strep-KU2, the function or the efficiency of the vaccine prevention is very high comparing with other vaccines. But I'd like to show you the pipeline of the general vaccine design that we developed for our cultures. In general, we should survey for the disease and find out which is the really infectious disease that cause the disease at that time. And after that, we should isolate the pathogens and characterize as a genotypic and phenotypic characterization. After that, we should clarify whether those isolated pathogens can readily cause the disease or cause a pathogenicity to the animals. After that, we develop the vaccine and test the efficacy and also the immunity response. So this is the overall pipeline of the vaccine in general. So let's go what we can make it for the vaccines. Let's start from the background of the Strep-KU1. We succeed to identify the pathogens from the disease fish. This is a characteristic of the Streptococcus fish, which show a bubbling eye or very big eye to the fish. And the fish die within a few days after infection. So we survey and list the distribution of the pathogens in the Thailand, in the northeastern, in the central, and also the southern part of the Thailand. And we can characterize two major pathogens, which is Streptococcus agalactiae serotype 1A and serotype 3, cause the majority disease in Thailand. For example, serotype 1A shown the red color here, serotype 3 the blue color here. But the distribution is really very different among areas in the Thailand. So we do further to determine the virulence of these pathogens, and we luckily can be conclude that it can be make a genetic correlation between the virulence genes. Here we using around the 16 virulence genes, and we found that those 16 virulence genes can characterize the virulent strain of the bacteria. And it's found that the serotype 3 has more virulence than serotype 1A after we conduct the pathogenetic testing in animal models. So from this data, we do further to produce the Strep-KU1 vaccine in laboratory scales. And we success to produce the vaccine that show the cross protection between serotype 1A and serotype 3. And this vaccine already tested in the field trial. And in the field trial, we already test in different areas of Thailand. We conduct for 4 years. For example, in 2015, in northeastern, in Nong Khai, in Kanchanaburi. And this is show how we conduct the injection of the vaccine in the farm trial. And from the result is shown here. This is a commercialized vaccine that come from the overseas and test the efficacy in Thailand around the 2015. It shows approximately 60% survival. Comparing with our vaccines, it shows approximately 90% survival in northeastern, and also the western area of tilapia cultivations. And then we conduct the efficacy again in the last year. It shows similar efficacy in Kanchanaburi and Antrong area. So we would like to conclude that the Strep-KU1 or inactivated vaccine against the Streptococcus in tilapia can provide at least 80% production yield when compared to the non-vaccine 50% and commercial vaccine 50%. So it means if you use the inactivated vaccines, you can earn or can increase the production yield up to 85%. But these technologies is ongoing to transfer the technologies to the animal productions company. But today, I'd like to introduce you about the platform product for producing the multi-molecular vaccine for aquacultures. Why this technology is important for aquacultures. Please come back to this diagram again. You're going to see that in the tilapia cultivation, it has a lot of disease, and some of diseases come in different times. So we need to have the vaccine that cover all of the disease that should be happening during the cultivation. So from this, we use the idea of multi-epitope vaccines to produce the vaccine that can be prevent the many species of the pathogen, different strain, and it don't need a different composition in the vaccine formulas. Let's come back to this technology again. For the subunit vaccine, we use these technologies, reverse vaccinologies, to produce the vaccines. And from the proteomics analysis and whole genome analysis of Streptococcus agaricales, we can make the comparative genomics and proteomics analysis. And we can take the antigenic proteins that found in both Streptococcus serotype 1A and serotype 3. And this antigenic protein that found in both serotypes is further used and further being as a subunit vaccines. This is show diagram how we can make the subunit vaccines. From this is show the diagram of the bacteria. It has the antigenic protein both in the surface proteins and also in the cytoplasmic proteins as identified by the genomics and proteomics analysis. If you use the whole cell vaccines to produce the vaccine as the inactivated whole cell vaccine, we call Sutkhet. But we already success to select only some of the antigenic proteins to be produced as the recombinant proteins and test for the efficacy analysis. We can make a Sutkhet. And this Sutkhet and Sutkhet, after they combine together, we can get the Sutkhet. But however, the Sutkhet and Sutkhet had a similar vaccine efficacy, but I'm not mentioned today. But I'm going to introduce you the further technologies that can make the vaccine more versatile and can make it more prevention to the different disease. We use the technology that can produce as a chimeric multi-epitope vaccines. Normally, the multi-epitope vaccine is normally used and made in the different to prevent the infectious disease, not only for the aquacultures. It's already in developed for the human being, but it's not successful to produce and use for control the disease. In the multi-epitope vaccine, mostly they combine different epitopes from different antigenic proteins. And although the epitopes come from the B cells or T cell epitope and link together with a short peptide linker like this. And because of the lack in vitro or in vivo analysis and no protein expression determination and no host immune response, so it's made all of the multi-epitope vaccine that report not functions. So in this work, we already proved that our vaccine can surpass this limitation. So we use the epitopes from the antigenic proteins to put and replace in the thin barrier structures of the proteins. These thin barrier structures show the epitope outside the core structure like this. It's like a propeller. And then these epitopes, we can put more, at least 5 epitopes to these structures. And this epitope might come from the different pathogens. And in this study, we use this epitope from protein called flavodoxin from the tilapia. And we on a plan to put the epitope from antigenic protein from the pathogens. So this is the pipeline of the primary multi-epitope vaccine designs. After we get the antigenic proteins that identify from proteomics analysis, we identify the epitope from each proteins and then subtract only the epitopes and put in the area of the thin barrier structure backbones. So we can toughen the epitope domains to many structures, and we're using the molecular docking analysis to search for the very good good 3D structures, and we can get the multi-epitope vaccine candidate. And from this analysis, we use the immunoproteomics analysis to extract the antigenic proteins, and we found the 79 proteins that be present in both serotype 1A and serotype 3 of Streptococcus lactiae. And out of 79 proteins, is characterized furthermore for expression ratios in the both bacteria. This analysis needs to verify whether all those proteins is expressed maximum quantity enough to be like antigenic protein candidates. After that, we get 5 antigenic proteins: BAC gene, RIP gene, CYP gene, CFS, and SPV1 genes. All those genes further characterized for all those epitopes. This is the list of the epitope that characterize from all those antigenic proteins. After that, those epitope is put to further analyze in the molecular modelings by put each epitope to the popular domains. And this is show the models of the epitope shuffling of the CMEV. You're going to see that we have more than 7,000 models of the epitope shufflings. And we success to make the very good promising tertiary structures from the molecular modeling. That is show the alignment of the epitope like this. We call 45F2 and 42E2 model. All of this is the peptide structures. After that, we make sure again whether these structures could be expressed well in the target cells. So we're using the in silico analysis to predict the protein structures. And we use Ramachandran plot to doing search whether the proteins is expressed in the allows secondary structure regions, and also characterized in another analysis to make sure that these primary proteins have the function as the antigenic proteins. And after that, we can conclude that this 45F2 and 42E2 is the best protein structure that can be used as a protein vaccine candidate. But we need to back translation to make this protein back to be the DNA. So we use reverse translation, use the codon optimization analysis and mRNA secondary structure prediction to make sure that this protein, after convert to be the DNA or mRNA, it can be expressed in the cells. So from this analysis, you're going to see the energies of these mRNA structures. It has very high negative energies. So it means this protein, this mRNA have the energies and the stability good enough to be expressed in the cells. After that, we make the recombinant protein vaccine and DNA vaccine. So we synthesize that sequence by using the nucleotide synthesizing. And upon this, so we can get 2 types of the vaccine. First is a protein vaccine, and the second one is the DNA vaccine. So in each vaccine, we have the 45F2 and 42E2. So let's go to see the efficacy of the vaccine after we doing the in vivo trial. But before going to the in vivo analysis, we need to make sure that our DNA vaccines can be expressed in the tilapia or in the fish. So we use the tilapia culture cell line to verify whether the DNA vaccine can produce. So this is the tilapia culture cell lines that infected by the DNA vaccine. We use the green forest protein as a tracking medium. So in case of the 42E2 and 45F2, after we doing the Western blot analysis, it showed the positive signal of the protein expression in the TK1 cells or tilapia culture cell line. And in case of the E. coli, this is the recombinant protein vaccine that we can produce. So in our hand, we have both the DNA vaccine and also the protein vaccine that can be readily used in the fish. So we verify further whether this protein vaccine and DNA vaccine can be prevent the fish from streptococcosis infection disease. So we separate to be the group of a negative control. For positive control, which is the benchmark, we use the formalin Q vaccines as the model of the vaccine and inactivated vaccines. And we have the DNA vaccine 42E2, 45F2, and we have the multiple protein vaccine E2 and F2 as well. So after we vaccinate the fish for 4 weeks, we challenge the fish with the pathogenic Streptococcus strain. And this is show the efficacy of the vaccine. This is show the relative % survival or how the fish survive after bacterial infection. This is the formalin Q vaccine, Streptococcus as a benchmark. If you see, it provides about 80% survival after this vaccine application. But in case of the DNA vaccine, it has no significant difference between the efficacy vaccine. So it means DNA vaccine, both 42E2 and 45F2, can provide the prevention as similar as to the Streptococcus. But in case of a protein vaccine, it reduces their efficacy significantly. So it's mentioned that this reduction might be come from the bioavailability of the vaccine. The protein vaccines might be clearance before the immunity is raised up, but the DNA vaccine can prolong the immunity because it needs time for change from the DNA to RNA and also the protein. So this is show how we can apply the DNA vaccine or protein vaccine in different stages of animals. And we confirm the vaccinated fish by using the titer assay. You're going to see that if you put the DNA vaccine and protein vaccine, the immunity of the DNA vaccine is raised up after injection of the vaccine, as well as the protein vaccine. But the immunity of the protein vaccine is decreased after 2 weeks. So it's well committed to the vaccine efficacy. So I'd like to conclude that about the vaccine, primary vaccine we named as the Streptococcus. It had the vaccine efficacy as similar as to the Streptococcus and other vaccines. In this, it might be show a bit low % survival, but actually this is very harsh condition because all of the fish die within 7 days. But actually, if you conduct in the field, this survival rate will increase about 80% because in the normal condition, the fish will not affect much and not rapidly die because of this disease. And this research is already published in a scientific report and have been invited to write in the book chapter. So you can read through the book chapter, Vaccine for Veterinary Disease, and we have 2 chapters here. But I would like to have more additional message about the alternative vaccine rules. As you see, the vaccine for the fish now is used the injection method. This is not usable much for the aquaculture because the fish cultivated in the water, so it's not easy to take it out. But now we can develop the vaccine is called Strep-R1. So you can mix this vaccine on the fish feed and give to the fish for at least 2 weeks. Fish will have the protection as similar as to the Streptococcus. Again, we also can produce another vaccine to prevent the Aeromonas disease, and also have the combo vaccine, Streptococcus and Aero disease, and also the viral disease, TILV. And together with the control of the disease, we also provide sensors for detect the disease in the field. We call the after sensors that can provide the on-site disease detection and also the antibiotic detections. So this is the final of our talk. So I would like to acknowledge the granting agencies at JICA to provide part of the financial support. The members from the Faculty of Fisheries, Tokyo University of Marine Science Technologies, and also NRCT, JSPS, and also DF. And this is a member of our team. So finally, I hope that our research can be support the SDG goal for the zero hunger, for the good health and well-being, and also the life below waters. So that's all of my talk. Thank you very much.
Thank you very much for your very interesting presentation. And now I think we have time for 1 minute for a short question. Is there any question from the audience? Okay, I have one question about the like half-life of the, I mean, vaccine when we inject into the fish. I mean, how long it will induce the immunity? I mean, and which technology is better?
The first question is how long that induce the immunities. For the one shot of injection, it can increase the immunity about 4 weeks. But if you put the second shot, at 4th week, the immunity is raised more until the 8th week. But in the field trial, only single shot can provide the protection for at least 120 days for 4 months. That's enough for the cultivation.
I see. Thank you very much. Is there any more question? Okay, if no more question, thank you very much, Associate Professor Dr. Sasimanat Unhachak.
Thank you very much.
Okay, I will move to the our next speaker, the final speaker today. May I invite Dr. Piyangpoon Leungchalernkit from Kasetsart University to present in the title of Light Regulation of 2 new manganese peroxidase encoding genes in Trimethyl's Polyzona KUI and WO27. So Dr. Piyangpoon, please start when you're ready. Okay, we can see your slide now.
Thank you for your introducing. Sawadee krap. Good afternoon, ladies and gentlemen. Today, I would like to present in the topic about the light regulation of the 2 new manganese peroxidase encoding gene in Trimethyl's Polyzona KUI and WO27. Light is an important environmental factor that regulates the fundamental process not only in phototrophic organisms like a plant, algae, and phototrophic microorganisms, but also important for the non-phototrophic organisms like a fungi. Fungi use the photoreceptor as an eye light sensor. Fungi use a photoreceptor like an eye light sensor to detect the light of the specific wavelength, such as the phytochrome for red light, opsin for green light, white color, and vivid flavoprotein for blue light. In the photoreceptor have a chromophore that's responsible for the specific light absorption, like retinol in the opsin and phytochromobulin in the phytochrome, and the last one is the flavin in the flavoprotein. For the example, the white color can binding to directly at the promoter of the DNA to induce the expression and regulation of the conjugation and carotenoid production in the fungi. Many biological processes controlled by light, such as the basidiocarbon formation of the schizophyllum commune, was induced by the UVA light, and the carotenoid production of the neurosporal casa was produced in the mycelium and conidia. And for the Botrytis, the production of the sclerotium in dark and produce the many spores under the light condition. And also the sporulation of the trichoderma was regulated by light control. In this research, we focus on the light regulation of the ligninolytic enzyme production of the white rod fungi, Trimethyl's Polyzona KUI and WO27. The ligninolytic system of the fungus, this fungus is composed mainly 2 kinds of the ligninolytic enzyme. First is the manganese peroxidase, and the second one is the lactase. Manganese peroxidase is a heme-containing glycoprotein that catalyzes the hydrogen peroxide-dependent oxidation of the manganese 2 ion to manganese 3 ion. Then the manganese 3 ion is compact with dicarboxylic acid for oxidation of the phenolic compound deriving of the lignin. And for the lactase is the benzene oxygen oxidoreductase or multiple copper oxidase that catalyze the oxidation of the many substrates and couple the reduction of the oxygen to the water. This fungus exhibits high activity of the manganese peroxidase and also lactase activity with efficacy on the dye and pharmaceutical product degradation, not only in dye reduction, but also decrease of the COD and also detoxification of the toxic aromatic dye. When we assessment with the duckweed or Limna minor and also mung bean Vigna radiata and bright shrimp Artemia salina. A few research focus on light regulation of the ligninolytic enzyme production, such as lamylase in 2010. Lamylase study on the effect of different wavelength of light on the lignin peroxidase production of the Phenol-Schistosporium. And they found that different wavelength of light had influence on the lignin peroxidase production. You will see that green light and yellow light have a positive effect on the lignin peroxidase production, while red, blue, and UV light have negative effect on the lignin peroxidase production. So for the objective of my research is to investigate the enzyme encoding gene in ligninolytic system of Thammitis polyzona KUIW027 and to study how they regulate enzyme production and enzyme expression gene under condition of illumination. This is of the overview of my presentation. We divide in 3 parts. First one is the cloning of ligninolytic enzyme encoding gene, and the second one is cloning of the promoter region of ligninolytic enzyme encoding gene, and the last one is the regulation of ligninolytic enzyme production and gene expression in different wavelengths of light. Part 1, the cloning of ligninolytic enzyme encoding gene. First of all, we designed the degenerate primer from the peptide fingerprint of the manganese peroxidate from Thammitis polyzona KUIW027. We select this region to design as a forward primer and this one for the leeward primer. Using design degenerate primer, 800 base pair fragment of manganese peroxidate gene was obtained by PCR. Then we sequencing this DNA, and the sequencing results show that 2 different manganese peroxidate genes were found in the Thammitis polyzona KUIW027. After that, we use the 5' rapid amplification cDNA and PCR to obtain the 3' region and use the 5' less PCR to obtain the 5' region. And then we combine the result to obtain the full length of mRNA. The full length of manganese peroxidase 1 consists of 1,331 base pair. And this gene shows 86% nucleotide sequence identity with manganese peroxidase 1 gene in the lens state Gibbbosa stain CD1. We also done with the manganese peroxidase 2 gene. The results show that the full length of manganese peroxidase 2 gene sequence consists of 1,306 base pair. The sequence of manganese peroxidase 2 show the 84% similarity with the manganese peroxidase 2 in Lensitate gibbosa stain CB1. This figure shows the phylogenetic tree was constructed using the deduced amino acid sequence of manganese peroxidase 1, manganese peroxidase 2 together with the other complete amino acid sequence of class 2 peroxidase, class 2 fungal cyclic heme peroxidase. The phylogenetic tree revealed that manganese peroxidase 1 here, and this one is manganese peroxidase 2 of the Thymitis polyzona KUIW027 are clustered within the subfamily A.2. The subfamily A.2 is the short tri-hybrid manganese peroxidase. Short tri-hybrid manganese peroxidase didn't have the activity of lignin peroxidase like subfamily A.1 and didn't have the versatile peroxidase activity like A.3. And because of the small molecule of short type manganese peroxidase, when comparing with the group B, and the group B is the typical long manganese peroxidase, this group have a larger molecular weight than the short tri-hybrid manganese peroxidase. Manganese peroxidase 1 and manganese peroxidase 2 of Thymitis polyzona KUIW027 had only 88% identity in their deduced amino acid sequence, suggest that they are distinct manganese peroxidase. For amplification of the lactase gene, we designed the primer based on the lactase gene sequence of the genus Thymitis and related species like Thymitis hirsuta, Thymitis versicolor, Thymitis gibbosa, and Thymitis also versicolor. After we amplify by using the design primer, we obtain the 1,200 base pair fragment of lactase gene by using the PCR amplification. We also use the 3' lase and 5' lase to obtain the full length of the mRNA of lactase 1 gene. The full length of cDNA of lactase 1 gene consists of 1,809 base pair. The nucleotide sequence of lactase 1 gene has 97% identity with the lactase 1 gene from the Thymitis polyzona MUL-T8443. The figures to show the phylogenetic analysis of deduced amino acid sequence of the lactase 1 gene was constructed with the author 35 completely amino acid sequence of the multiple copper oxidase gene from the different sources, including the Basidiomyces lactase, plant lactase, Ascomyces lactase, insect lactase, and also the bacterial multiple copper oxidase. This result reveals that the lactase 1 gene is clustered in the Basidiomyces lactase at the same branch of the lactase from Thymitis polyzona, shown in this one. So for the part 2, for the cloning of the promoter region of ligninolytic enzyme encoding gene, for obtain the promoter region of the ligninolytic enzyme gene, we use the inward PCR. In the inward PCR, we extract the genomic DNA and then we digest the genomic DNA by using the restriction enzyme like KPN1, BAMH1, NCO1, XBAR1, XPA1, XPAL1, PGL2, EcoR5, and the mixture of the BAMH1 and BGL2. After we digest with the restriction enzyme, we cell ligase the template and use as the template in inward PCR. After we amplify the fragment from the inward PCR, we sequencing and predict the promoter sequence and the cis-acting element in the promoter region. Figure 3 shows the promoter region of the ligninolytic enzyme encoding gene with the cis-acting element in the promoter. The promoter region of manganese peroxidase 1, manganese peroxidase 2, and lactase 1 show low sequence similarity with those of the other fungi, and we found the unique sequence in their respective gene. However, all promoters share the common regulatory region, such as the metal responsive element, this one metal responsive element, and the xenobiotic response element shown in green. And the last one is the heat shock element shown in blue. This one is the common regulatory element that found in the ligninolytic enzyme system of the manganese peroxidase 1, manganese peroxidase 2, and lactase 1 gene. And concept sequence of the Tata box, this one, and inward cat box and the cat box is the were found in core promoter region. Interestingly, we found the light response. We found the light responsive element in only manganese peroxidase promoter MMP1 and MMP2, but not found the light responsive element in the lactase promoter. This one, the occur of the light responsive element may be these two genes will drive by the regulation of light. So we will do the part 3 is the regulation of the ligninolytic enzyme production and gene expression by different wavelength of light. For the preliminary study, we culture the Trametis polyzona KURW027 in the potato dictose agar. We supplement the substrate of the enzyme into the potato dictose agar. We use 2,6-dimethoxyphenol at 1 millimolar as a substrate to supplement in the potato dictose agar. When the when Trametis polyzona produce the ligninolytic enzyme, the ligninolytic enzyme can oxidize the substrate 2,6-dimethoxyphenol and resulting in the orange color that show in figure A and B. In the figure A and B is different one of the light condition. The first one is the fungi is cultured in the potato dictose agar supplement with 2,6-dimethoxyphenol in that condition. And the second one is the potato dictose agar supplement with the substrate of enzyme in light condition. And the last one is the control. We culture the fungi in potato dictose agar under light condition to confirm that the orange color that produced is from the enzyme activity, not produced from the pigmentation of the fungi. In the result, you will see that the activity of the enzyme is increased under light condition, while no observe the difference of fungal growth between light and dark condition. The growth is the same. So we would like to study the regulation of the enzyme production and enzyme expression in the liquid medium. So we prepare the inoculum. We culture the Tyrosine polyzona KUI-W027 on the potato dictos agar and use a 5 apical hypothesis used as the inoculum in the 50 milliliters of colored medium. Then we shaking at the 130 RPM for 3 days. And then the mycelium were washed and collect and use as the inoculum in the new medium of the colored medium. For this one, we culture in the colored medium with the exposure with the different wavelength of light. At the light intensity is 500 lux with the tungsten light and blue LED with the specific wavelength at 465 nanometers, and the culture in the dark we was used as the control. After that, we assay the activity of manganese peroxidase and lactase activity, and we use as the 1 unit of enzyme was defined as the amount of enzyme that caused the 1 increase in the absorbency per minute in assay condition. And the last one, we collect the mycelium and extract the RNA to evaluate the enzyme expression by using the real-time PCR. Figure 5 shows that the effect of different wavelength of light on the ligninolytic enzyme production and gene expression in Tyrosine polyzona KUI-W027. You will see that the tungsten light increase the manganese peroxidase activity that shown in red bar. The activity of manganese peroxidase was increased up to 1.5 times. You will see that the level of manganese peroxidase and lactase activity in dark condition was 2.2 and 1.9 unit per ml. And when we culture the Tyrosine polyzona in the blue light, you will see that blue light is decreased the enzyme production both of the manganese peroxidase and also lactase. When we focus on the enzyme expression, you will see that only tungsten light upregulation of the manganese peroxidase genes, both of the manganese peroxidase 1 gene and manganese peroxidase 2 gene, but not upregulate of the lactase 1 gene. Manganese peroxidase 1 and manganese peroxidase 2 was increased up to 6.5, 6.5, and 3.8 fold of induction. And you will see that no significant difference of the 3 gene expression was observed under dark and blue. The expression level is the same, but the activity under the blue exposure was decreased. So we think about maybe light, blue light is important to the stability of the enzyme. Compared to the equivalent dark control, the both manganese peroxidase and lactase activity were relatively stable under tungsten light. In the first 6-hour exposure of the tungsten light shown in yellow line. For the first 6-hour exposure, the retaining of the 90% to 95% initial activity was observed. Conversely, over the same time course, the blue light have more stronger to the negative effect on the activity of enzyme lactase and manganese peroxidase. The lactase and manganese peroxidase will retain the activity of both enzymes, falling at the only 70% after exposure with the blue light. For the manganese peroxidase activity, retain activity progress decline to the 39% after exposure with the blue light at 72 hours of exposure with blue light, whereas the lactase activity retained only 6% after exposed with the blue light. So this one confirmed that the blue light is involving in the enzyme stability of both enzymes, activity of the manganese peroxidase and also lactase. In the conclusion, the ligninolytic enzyme system of Thymitis polyzona KUIW027 composed of mainly two kinds of manganese peroxidase and lactase encoding gene. Reduced amino acids reveal to manganese peroxidase in Thymitis polyzona as a new member of the class 2 fungal cyclic heme peroxidase. Metal responsive elements, xenobiotic response element, and heat shock element were commonly cis-acting elements found in all ligninolytic enzyme encoding gene of the Thymitis polyzona KUIW027. Interestingly, we first found light responsive element on the promoter of two new manganese peroxidase in Thymitis polyzona KUIW027. Due to those regulation elements, like ligninolytic enzyme production and gene transcription in Thymitis polyzona KUIW027 could be regulated by light. And blue light is effect or involving on the ligninolytic enzyme stability. Finally, we would like to thank the Department of Microbiology, Faculty of Science, and also Kasetsart University, and the Laboratory of Applied Microbiology Graduate School of Bioresource, Miek University, and also thank to the Royal Golden Jubilee PhD Program, Thailand Research Fund for support my PhD, and also thank to the Kasetsart University Research Development Institute. And I would like to thank all of my supervisor, Associate Professor Dr. Lerlak Chitdon and Assistant Professor Dr. Churapha Thirapasakul at the Department of Microbiology, Faculty of Science, Kasetsart University, and also thanks to the Professor Dr. Kazuo Osaka and Professor Tetsuya Kimura, Dr. Makiko Osaka, and also Assistant Professor Dr. Emi Kunetake at the Laboratory of Applied Microbiology, Graduate School of Bioresource, Miek University. Thank you for your attention.
Okay, thank you very much. So we have actually a little bit, maybe one question from the audience. Is there any questions? Okay, if no questions, thank you very much, Dr. Priyangkun Chalankit, for your interesting presentation. So today the meeting is, the time come to an end, and I am sure that we all learned so much from our interesting presentation today. So may I next invite Assistant Professor Dr. Sutsai Sin Kaew Leung, Vice Dean of Academic Affairs, Faculty of Agriculture, Kasetsart University, and also the chairman of this symposium, to deliver a closing remark. Please, Assistant Professor Dr. Sutkhet Kaewlua.
Distinguished speakers and chairperson, honorable participants, ladies and gentlemen. The symposium is about to come to an end, and I have been asked to say a few words on behalf of Kasetsart University. In my honest opinion, this has been very successful Congress. The discussion, the ideas shared have been warmly received and are appreciated. I'm sure the existing bond of friendship and cooperation between our university will continue to flourish in the year ahead for our mutual benefit. On behalf of Kasetsart University, I would like to express our most sincere thanks to all the speakers. The research outcome and project that you presented provided us with an invaluable opportunity to learn. I know that all those who participate in the program found it extremely beneficial. We all have greatly appreciated an opportunity to exchange perspectives. Also, I would like to thank the committee members, chairpersons, and participants for your valuable presentations and fruitful discussions. Finally, I wish to express, appreciate, and thanks the entire organizing team for creating this virtual convening. Ladies and gentlemen, it has been a long but rewarding day. May I now reaffirm my appreciation for your valuable support, and I look forward to many more years of creative, successful cooperation. Thank you. Before we leave this room, I would like to ask you to complete our assessment form, evaluation form. You can go through the QR code here on the screen and then you can just take a few minutes for complete. Thank you again and goodbye. See you next time. สวัสดีครับ
So the link for giving the feedback also on the chat board. If you are convenient, you can click on the URL link on the chat board and also give us some feedback. Thank you.